Hello. It's my great pleasure to introduce the International Symposium about Genetic and Artificial Intelligence in Buenos Aires, Argentina. First of all, I want to say thank you to the full support of the Argentinian Council of Ophthalmology, the Latin American Society of Image and Artificial Intelligence, the Buenos Aires University, the FASTA University, and Maimonides University. Also, we working with an international coordinators very well known around the world in the field of the genetics and artificial intelligence. From Argentina, Professor Chiara Dia is a full professor of Buenos Aires University and the head of the ophthalmology department of the Buenos Aires University Hospital. John Packrash Gian is, as you very know well, one of the key leaders in genetics fields, working in a National Health Institute and the International Consortium of Genetics in Ophthalmology, and also is the editor of the series well-known books regarding investigation and advance in ophthalmology and many, many other functions. Natarajan is, well, a specialist in retina from India, and they was a president and are involved in a lot of different societies around the world. Is the General Secretary of International Council of Ophthalmology, also was past president of the All India Ophthalmological Society and Trauma Asia Pacific Society, and also the International Society of Ocular Trauma and many others. And Amit from Canada is from the Waterloo University, but also they is part of the lab, and also is the CEO of software and sequential company, and is very well working in artificial intelligence, not only in ophthalmology, also in medicine. And it's for me a great pleasure all this group of ophthalmologists working together to organizing the symposium. And we have other key leaders in the world, and I made a presentation of the first presentation from Dr. Gian Packrash from the National Health Institute, and the topic will be the international programs and opportunities with Global Eye Genetics Consortium. Thank you.
I guess you can all hear me. So good afternoon from Buenos Aires, Argentina. It's wonderful to be here. What a great honor to be actually working with the Latin American team, President of Argentinian Council of Ophthalmology, Professor, and of course, all the distinguished friends here. Also very important, special thanks to our host, Professor David Pelagius, who's just been an incredible host, organizing this in such a short time. So, bravo, David. I think that's how you say in Buenos Aires, right? So, bravo, David, right? And so I bring greetings from the National Eye Institute Director, Dr. Michael Cheng, and we are very excited to be really working in Latin America with scientists, a totally new experience. But let me just quickly go through and give you an introduction of what National Eye Institute has done. So let's see. Okay, so no financial disclosure, and opinions expressed here are my own, not of the government of the United States, so the government doesn't take any responsibility. Really quick. So National Eye Institute. So how many of you have heard of National Institutes of Health? NIH. A few, okay, a few. So National Institutes of Health is the largest biomedical organization in the world, right, with a budget of close to $48 billion. So that's the big number, right? So wherever that all that money goes, it decides really the current and the future of biomedical research around the world. So it consists of 27 institutes and center, as you can see here. Okay. So the star over there, I guess yeah, pointer is working. So National Eye Institute is one of the mid-size institutes, and we call it here in Argentinian language jumbo shrimp, right? So it's like a decent-sized institute with a budget of close to about $895 million a year just for eye research. Amazing, right? But we do a lot of work, and we get to work with people around the world. And our mission is essentially, let's see. Okay, so if you look at this, the number of foreign funded, and we work with scientists around the world. So number of foreign NIH funded direct awards and collaboration in the World Report. You can go online and see it in the World Report. And these are the countries that you have 100 plus records from last year. And you can see there are many, and Dr. Amit Mathur is here. You know, of course, Canada is a very important partner. Maximum number of foreign awards go to Canada. But if you look at the list here, these are the top, about 90% of our records in these 42 countries. And Argentina is, of course, you know, one of them. But there are many Latin American countries in here. And Argentina is, what is it? Right here, right? So there are many Latin American countries. So we are already working with you, but we just want to actually, you know, expand our offering and work in new areas of research. Okay, so now coming to the National Eye Institute, research with a foreign component. As you can see that in the last 7-8 years, our business has grown, our collaboration has grown from 28 countries to close to 60 countries. And these are the unique projects and investigators and working with the programs, in the programs with about 60 countries around the world. And here's that many of us have flown in here, come from different places around the world. So here's a landing map. As you can see that, you know, we are right up north from you. So there are many programs, many projects that are being worked here in your region in Latin American countries. Okay, so what is important here? That we have a new strategic plan, and this was developed just a couple of years ago. And I have a QR code, and I would highly encourage you to take a picture of the QR code, because that will actually give you a very comprehensive view of the National Eye Institutes and deciding, working, and really developing programs for eye research, working with scientists around the world. And these are on the right-hand side, you see the traditional areas of eye, diseases, retinal diseases, corneal lens, glaucoma, strabismus, and so on, low vision. But our new strategic plan, puts emphasis on the 7 cross-cutting areas. So the cross-cutting areas you have, from genes to disease medicine, that's the genomics, working on genetic aspect of it, biology of neuroscience, immune systems, regenerative medicine, data science, AI, that they are very, very important. And this, again, goes into all areas of eye disease, quality of life in the public health program. So this is really a document that I would highly encourage you to actually look at and study. So why do we pursue international collaborative research? These are the main reasons why we go and work with the scientists around the world. Unique populations here in Argentina, in Latin America. You have unique populations. So genetics is really one of the top reasons, actually. Unique disease presentations, epigenetic factors, unusual exposures, infections, diet, cultural context, so on. This all seem to have some effect, some relation somewhere with eye diseases people have reported from around the world. Unique interventions, working on technology, novel technologies, frugal technologies, how to do science, how to do ophthalmology in a much more cost-effective way. Mobile health, that's very, very important. So these are all areas, alternative health system, data science, large data set, working with all of you, AI and data using mobile technology, diversity, working with researchers around the world. So that makes it a very impactful research. Okay, so what are the recent global programs that you can pay attention to? So you can actually look at that we are working with a large number of principal investigators, PI, and institutions around the world. And one of the things that when I tell people that there are actually anyone at an institution or organization from any country around the world can apply for NIH grant. People don't know that. People are very surprised. So you don't have to be an American citizen in order to actually apply for American grant. But usually if you're doing it first time, it's better to work with somebody who already has a grant, somebody who knows the art of grant writing, because art of grant writing is not only science, but it is also an art. That's something that people have to perfect it. So you can work with an American PI, principal investigator, in order to get started. But anybody from anywhere in the world can actually apply for an NIH grant. And then if you look at, there are different kinds of programs, government-to-government collaborations. We have a lot of training programs, short-term, long-term training programs open to all. We have had a lot of Latin American scientists working with us. They come for training. They come actually work in we have 20+ labs, and they come and receive their training. A lot of Brazilians have received training, Argentinians, and so on. So, and then they have come back and they have set up their own lab. I mean, Dr. Rubens Belfort, he was trained in Brazil. He was trained at NEI, came back to Brazil and in Sao Paulo, and developed a huge program of his, in Colombia, and so forth. There are many organizations. There are actually two brand new programs that we are introducing now. One is called K01 Grant Program. That's the International Research Scientist Development Award. You can apply for that. Oh, sorry. Okay. Can you hear me now? Okay, much better. So sorry. So there are two, so important thing, two new programs that we are announcing. And here I announced at ICER, which just took place a few days ago. Here, K01 grant program, take a look at it. That's many Latin American countries actually qualify for low and middle income countries, which is LMIC. All right, same here, K43 program, that's for Emerging Global Leader Awards, LMIC, okay? So if you're interested in, the details are there on our website, but you can reach out to my colleague, Dr. Niraj Agarwal. That's his email address. Please feel free to take a picture. So in addition, we have several special initiatives, several programs. I would like to talk to you about just one program which we talked about here is Global Eye Genetics Consortium, which is a consortium that we develop bringing international scientists from around the world. It started about 10 years ago at NEI, working with Tokyo Medical Center, and now we have added and brought 36 countries working with, and you'll hear more from Dr. Takeshi Iwata in one of the talks right after me. And the other one is that National Eye Institute is bringing all the top 10 societies from around the world, like American Academy of Ophthalmology, APAO, ARVO, ICER, RANSCO, IAPB, ICO, WHO, together to really work on many global problems. And these are the committees that we have currently. These are the areas we are working on: global training and education, research partnership, and standardization of methodology and creation of global data standards. As you can see that we are co-chairing these programs. For the first one with AAO, American Academy, the second with ARVO, and the third one with APAO. So, and this is our last meeting at ARVO, just a few months ago we had, and this is a global committee that works with us on all these programs. So hopefully we'll be able to actually make some milestones. This is, again, on the Global Eye Genetics Consortium. You can see that this was founded about 10 years ago and really made a great stride into developing, bringing all the genomics work under one roof, under one programs. And currently, we have more than 200 members in about 30 plus countries, 38 countries. And in just last 8 years, forming last 10 years, we've produced 4 volumes of Advances in Vision Research published by the Springer Nature. And the chief editor is, of course, Dr. Arun Singh, who is very good friends with Dr. David Pelagius. So here's my contact information. And thanks to Michael Cheatham and my colleague who has provided a lot of data for my talk. And thanks, Michael. I know you'll be speaking soon. And also thanks to Dr. Takeshi Iwata, who is the current president of GEGC, and he'll be speaking shortly. And here's my contact information. Again, thank you so much. And it's a pleasure to co-chair the program with Dr. Pelagius here. And appreciate and I'll be around all day for any informal discussion.
Thank you, John. And for me, it's really an honor to be part of this meeting. And I'd like to introduce Dr. Iwata Takeshi, who is PhD, and he's the head of the Molecular and Cellular Biology Division in the National Institute of Sensory Organs and National Hospital Organization Tokyo Medical Center. And he's going to talk about addressing eye genomics research with global collaboration. Thanks.
Hello, my name is Takeshi Iwata, the director of the Molecular and Cellular Biology Division at the National Hospital Organization Tokyo Medical Center. I'd like to first thank Professor Peles, Professor Prakash, and Professor Natarajan, and Professor Jaladia for inviting me to this meeting on current challenges in eye genomics research in Argentina and Latin America, a collaboration with National Eye Institute, NIH. I have no financial disclosure to declare. I'd like to start with this slide showing a double-stranded DNA wrapped around histone molecule to create a nucleosome, further packed into chromatin and the chromosome, and then into the nucleus of the human cells. Any mutations or nucleotide variants can be a cause or a risk to eye diseases. When the eye disease is caused by strong genetic factors, it's called inherited eye diseases, such as retinitis pigmentosa and macular dystrophy. On the other hand, when there's other factors involved, such as environment or behavior, it's called the common eye diseases, such as AMD, glaucoma, cataract, and myopia. This slide shows the different inheritance patterns of genetic diseases. To the right is the autosomal dominant. The father has one of the chromosome mutated, inherited to his daughter, and then further to his grandson and granddaughter. To the left is the autosomal recessive. One of the two chromosome is mutated and becomes a carrier, and when the son inherits to one of the mutated chromosome from father and mother, he gets the diseases. But if you inherit only one of the chromosome with mutations, it's normal phenotype. Here is the X link in the middle. The father has only one X chromosome, which is mutated. And when it's inherited to his daughter phenotype is normal because she also has a normal X chromosome. But if it's further inherited to his grandson, the grandson will only have one X chromosome mutated, and he will have the disease. One of the important group of inherited eye diseases is called inherited retinal diseases. It's about 36 different type of inherited eye diseases called the retinitis pigmentosa, Stargardt disease, choroideremia, occult macular dystrophy, X-linked retinoschisis, and best diseases. Since the 1990s, over 300 genes has been isolated and identified, and it's registered in a database called RetNet. However, all this information comes from a patient in developed countries. To the left shows the Asian human migration, which started 200,000 years ago to 12,000 years ago, as showing a Homo sapiens moving toward Europe and to Asia and all the way to the American continent. To the right is a modern human migration of 2024, reported by United Nations, showing millions of people moving around the globe, continent to continent. Most of the eye diagnostics can be available in the United States and countries in Europe and countries in the Pacific Rim. However, majority of the population exists in Asia, in Africa, and South America, which is untouched. So in 2014, we started a research consortium targeting countries in Asia, Africa, and South America to promote research in eye genetics. We usually start by networking and research collaboration, publication, and research grant submissions. Since many of our members do not attend major meetings like AAO or WOC, ARVO, or ICER, we go to each of the countries to promote eye genetic research in their local ophthalmology meetings. So since 2014, we have been to many countries around the globe, and just recently we went to the country in Africa. 2 months ago, Dr. Prakash and I attended the Africa Ophthalmology Council, which took place in Kigali, Rwanda. About 300 people attended our GEG sessions, and we had a very good discussion about the situation in Africa. GEGC has two publications. One is the newsletters, which is published every month, and also a book, which is published 2 or 3 years. So this is the GEGC Global Network. Starting from 2001, the Japan Eye Genetic Consortium was established. 2013, Israel IRD Consortium was established. And then China Eye Genetic Consortium, followed by India Eye Genetic Consortium. And I see the Pan-American IRD Consortium established in 2019. And then in 2023, Indonesia Society of Genetic Eye Disease and Cell Therapy Working Group was established. And this year, we established the African chapter of GEGC. And these are the members actively working around the globe to promote eye genetic diseases in Asia, Africa, and South America. Since we have established the global network, now it's time to use this network to start working on research. So one of the examples that we can follow for GEGC is the Japan Eye Genetic Consortium, which we follow the next 7 steps to the clinical trials. So the most important step is the patient diagnostic collection of phenotype of information. So Department of Ophthalmology in universities or eye hospitals or eye clinic information will be uploaded. Retinitis pigmentosa, Leber congenital amaurosis, macular dystrophy, or station 99 blindness information will be uploaded. For each diseases, there'll be a specialist who monitors the incoming information and corrections if needed. And once this phenotype is corrected, then it will go to the whole genome analysis, and the result will come back to the database, which the university and the eye hospital can observe and to return the result to the patients. So here's the phenotype genotype database, and you can see the server here, fundus photo, the visual field, the fluorescence, angiograms, OCT, and ERG, and also the text information is entered into this database. The third step is collecting the samples. So there's two way of collecting this samples. One is the saliva, which is very easy because you can collect in 2 milliliters of saliva into this tube and flip this cap, and it will be mixtured with this special reagents that can stabilize the DNA for about 1 year in room temperatures. However, this DNA will contain some of the microorganisms exist in saliva, so it's not suitable for whole genome sequence. However, for whole exome sequence, it's a very good system. On the other hand, you can collect the blood, which the patient needs to go to the hospitals, but if you store it in -20 degrees, it's extremely stable. There's no microorganism contamination in this blood, so you can do straight to the whole genome sequence. The DNA extraction usually can be done manually in the lab, or you can use a robot like this, but we suggest that you outsource for DNA extraction to a company. If it's eluded to a TE buffer, it can be a very short-term surge. But if you precipitate in ethanol, you can store it for a very long time. So next step, fourth step, is the whole genome analysis to identify genome mutations. There are currently two methods to do the whole genome sequence. One is the short read, and the other is the long read. The short read is to fragment the genome into very small piece and just reading 150 base pairs of the both end of this fragment. It is very relatively affordable, but some of the piece, because this is short, cannot be identified where this piece is coming from the genome when it's mapped. On the other hand, there's a long read, which reads about 20 fatal kilobase, overlaps very well with the other piece to make the good genome sequences. So once sequence is done, we need to use this analysis of a pipeline to identify the mutation that is causing these diseases. So here you see the pipeline that we established. Took about 3 years to create. And here's the phenotype database, and this is the sequence server. And then you can see 60 servers for analysis. The fifth step is to prove that your mutation is actually causing the disease. And this requires functional analysis to elucidate the molecular mechanism of disease onset. And currently, we have many tools to do this. First of all, the next generation sequence actually does not only sequence DNA, but also RNA, methylated DNA, and also identify chromatin structure analysis. Also, we have iPS cells. We can generate iPS cells easily from blood cells and differentiate them to RP cells, retinal ganglion cells, or photoreceptors, and also differentiate retinal organs if necessary. Also, we have a gene editing technology like a CRISPR-Cas9 to create a knock-in models, animal models. You can also edit the iPS cells and also use this as a therapy to correct the gene mutation in vivo. Also, the advancement in the mass spectrometers to detect protein, small molecules, detect protein modifications, and detect protein expression levels, and also detect protein-protein interactions. So once the molecular mechanisms of disease onset is identified, there is many opportunity to develop the molecular therapy. So the sixth step is development of molecular therapy based on the basic information of disease onset. So this is one of the example of a dominant, autosomal dominant, inherited normal tension glaucoma pedigrees. So we made iPS cells and differentiate them to neuronal cells. As you can see in the patient cells, the protein is aggregated, while in controls, it's relaxed. The protein is relaxed. So we assume that there is some kind of a protein-protein interaction abnormalities in these patients. So we looked for this protein, wild-type proteins, and then we search for protein that is interacting. We found a normal protein interacting. But in the mutant proteins, we see something else binding. In this case, mutant optineurin was binding to a protein called TBK1. So we figured that if you inhibit this interaction of mutant protein with a TBK1 protein, we might be able to reduce the phenotype of glaucoma. And exactly what we did. We used the FDA and PMD approved inhibitor, the TBK1 inhibitor called amzenonox, into a knock-in mice, which was created by gene editing, and applied for about 12 months, and showed that this inhibitor can significantly reduce the degeneration of the retinal ganglion cells. So step number 7 is the last step, the clinical research and clinical trials. If you have been doing from step 1 to 6, you may have some therapeutic systems that you have developed who want to try for clinical research. But also, you may be asked from companies to do the clinical trials. So in Tokyo Medical Center, we have been doing RPE65 clinical trials and also RPGR clinical trials and for inherited retinal diseases. Also, inherited retinal diseases panel systems has been also been done in Tokyo Medical Center. So you have seen a step number 1 to 7 of the Japan Eye Genetic Consortium. GEGC is currently step number 1, but we are eager to start a project of collecting samples. So our Vice President, Im-Bing Hu, from Baylor College of Medicine, has assembled this team from USA, South Africa, Nigeria, Indonesia, India, and Nepal to conduct a research. And we want to submit this application to NIH. Thank you once again for inviting me to this meeting. Participation of Latin America is critical for GEGC operations. Please visit our website and participate in our activities. Thank you so much for your attention.
Thank you, Dr. Takeshi Iwata. For such a detailed description of GEGC. I'm sure a lot of people are very excited about it here in Latin America. So it's a great honor and pleasure to introduce my colleague, Michael Cheatham, who works with me and rest of the NIH. He's at the National Institute of Health Office of Director and in charge of World Report. Before coming to NIH about 10 years ago, Michael had actually done a lot of work in international partnerships in distinguished organizations such as AAAS and so on. So he's going to be talking about really a very important topic, which I love, that is really, how to work with, how to find good collaborators, how to work with people around the world, actually investigators who have the right approach. So it's a great pleasure to introduce Michael Cheatham. Michael, take it away, please.
Thank you very much. Can you hear me okay? Oops, you've just left the stage. I'm assuming you can hear me okay. So thank you, John, and to the organizers for the opportunity to introduce World Report. I will, let's see if I can get my slides to move. I'll give you a brief introduction to what it is. I'll tell you how it's being used, and I'll end on where we hope to go in the future with it. So World Report is an atlas of health research investments. It's hosted at NIH, but it's data from a partnership of 17 health research funding agencies. And together, the data set comprises about 1 million direct records of research awards and collaborations in 187 countries. Over 28,000 research organizations are captured, with about 170,000 unique investigators in the system. It is free. It's available to the public. That URL there works for the most part. It's not great on mobile. It's a legacy app. but it is pretty good on the desktop, and it's very, very easy to use. So about 10 years ago, the idea came at a heads of international research organizations meeting, HEROES, that could we build and collaborate on a database on global health research in Africa? And somebody volunteered to pool our funding data and came up with this prototype of World Report. And the initial use case was, can we use this platform to analyze and understand the research landscape? And can funding organizations identify gaps and duplications in funding and perhaps actually synergize those investments? Fast forward about 10 years, and World Report with the same data is now looking very much like an opportunity to do portfolio analysis, to identify and strengthen international collaborations and global research networks. And then a topic that's very close to my interest, helping investigators globally find potential collaborators as well as targeted funding opportunities. So I'll explain a little bit. Why did NIH need to create a new platform to do this? There's a perfectly good inventory of systems that NIH uses. NIH Reporter is the official record of investments. So I took a look at Reporter, and a very simple high-level search of NIH in Brazil for 2023 generates 9 grants to 5 Brazilian research organizations, and you can see them there in those red boxes, and you can duplicate this research. The same query in World Report gives you a very different picture of NIH and Brazil. So here you have 250 unique research projects funded by NIH that are being carried out through 500 collaborations at almost 180 plus research organizations in Brazil. And I've just grabbed a couple of the screenshots there to show you what you see when you go into the app. As John was saying, NIH is somewhat unique in the federal government in that it can support the best science anywhere, and it does. So in 2023, NIH made a little over 500 awards to foreign research organizations in almost 60 countries. However, as John accurately expressed, the real international action is in research collaborations. And here we have over 21,000 research collaborations in 161 countries. It's much easier, as he pointed out, to form an international research collaboration, to get engaged with NIH, than it is to go in and write your own application, at least at the first instance, the first time you've worked with NIH. So how do we use this thing, this World Report? It's exceedingly simple. The app has visual summaries of data. The numbers that you see are the numbers of research organizations in that country. The app generates a little breakdown of whose records you're looking at. So NIH is the largest share of records, but then the Canadian Institutes of Health Research are about a quarter of those records. It breaks them out by geography, and it gives you a little snapshot of the top 10 most prolific research organizations in the data. The user experience is so easy, I don't need to explain how to use it. So you can just poke around on the map, or you can work through the bottom half of the screen, the results pane. Here's a snapshot of South America for the last 2 years. And if you drill down into Argentina, you see that there are 377 instances of NIH research collaboration in this time period. Sorry, this is not just NIH, this is all World Report funders. This is 5 funders. Over 200 unique research programs at 144 Argentine research organizations. And here's the breakdown again. About 3/4 of that activity is NIH, but the rest is from a variety of funders. Again, any of the filters on the left side of the screen will get you to where you're going, and then the bottom half of the screen populates. What's really powerful, however, is that all of the data is freely available for export. So that initial use case, World Report developed for research funders to do strategic cooperation and now portfolio analysis, is very much something that happens on a routine basis at all of the partner agencies. However, the community researchers is using World Report to build out these research collaborations, because you can find things very easily and you can map health research in a usable way and find collaborators. Finding project funding is something that is a new use. I would call it an off-label use for World Report. It certainly wasn't designed for that, but it's actually quite good at it. And I'll walk you through the 3 simple steps for how you use World Report to identify NIH funding opportunities. So go back to your filters and find similar funded work. It can be anywhere you want. Make a note of the PI and any of the collaborating institutions. In the app, solid dots are direct awards, and hollow dots are research collaborations, and then the half and half are both. So once you find a research activity of interest, click on the program title in World Report, and it's a hyperlink back to NIH Reporter. And here you'll find additional details. The most relevant for this audience are the program official and the funding, the specific funding opportunity. Now you know exactly who to talk to about your research interest because you can see everything that's been funded through that particular program. And finally, in Reporter, you can look at similar projects, copy and paste these research titles or the PI name back into World Report. And there you can see where there are international research collaborations on those activities. So again, it's not designed to do this, but it actually does a pretty good job, and it's pretty easy to navigate back and forth. When you export the data, our users do things like this landscape analysis of the research seen in Africa for a year, and then they drilled down and they looked at an institutional level analysis to see who was funding which institutions most frequently. And finally, they looked at funding agency responses to topical events. So in this case, it was the 2014 Ebola outbreak, prior to which there was very little Ebola work being funded in Africa, but that ramped up quite quickly in the subsequent years to that outbreak. And finally, this is the last one. This is an overlay of the global burden of disease on the research portfolio, again, in that same region, to see whether the funders are really speaking to the needs of the country or region. Another World Report use that I won't really go into, except to just flag it here, we are partner with the WHO and the Global Observatory on Health Research and Development, and we're using World Report as part of a proxy framework to assess health research capacity. This framework uses 2 inputs, research funding and number of PhDs, health PhDs awarded in a year, and 2 outputs, publications and clinical trials registered as a synthetic framework for assessing research capacity and targeted interventions. So what do I do now? What's the future of World Report? World Report administratively moved within NIH and is now integrated with an office in the Office of Portfolio Analysis in the Office of the Director. And here we're looking at things like tracking and predicting the impact of NIH decision-making using tools that we develop in-house. Tools such as the relative citation ratio, which normalizes across different fields. It's a much more responsive metric than some others in use. We're looking at deep learning analysis of investments and publications. There are tools that assess the rate of scientific progress and emergence of new ideas. And then there's finally a tool that is the approximate potential to translate that can be applied to both an awarded grant or to an application. The Office of Portfolio Analysis, as its name implies, does a lot of comparisons between different funding organizations to see where there are overlapping interests. This is a zoomed-in view of a portion of the behavioral science portfolio of the NIH as compared to the US National Science Foundation. This can be done through World Report data as well among those 17 funding agencies. I mentioned emerging fields and rate of progress. This was part of an analysis done for NEI a few years ago to see which new topics in eye research were being funded by NEI. and then how those topics perform compared to the rest of the field. And finally, we can use World Report data to track collaborative networks, to see where the patterns are, and to see where funding agencies can actually help strengthen those connections and provide opportunities. So why am I here? Because I would like our partnership to grow beyond our 17 members. We do have 5 new members joining by the end of this year. We're also looking at public-facing data resources where we can scrape data and incorporate it into World Report. We're targeting countries with significant health research capacity and countries of strategic importance to health research. We think we're useful not only to the funding community or to the international lending community, but we're also useful at the research organization and in academia. Finally, development agencies, foreign ministries, and even civil society have developed applications on this data, which is very simple. It's just the project, the institution, and the investigators. And there are 12 essential items, including start and end dates, things like this, an abstract. There are 10 additional data fields that we incorporate as available. And the closer we get to full reporting from all our partners, the more robust the platform is. So I would make the case now in Argentina with everything that's going on with the CONICET, it is an ideal time to bring a new partner into World Report and to share with the world what's being funded in Argentina and how do we put this research on the map and integrate it into the system. With that, I will end. Thank you very much.
Thank you. You have a microphone, please. Yeah, thank you very much, Michael. Really appreciate it. This is just wonderful. So I was just wondering if we have him here, and certainly he's not going to be joining us. He's enjoying his garden, as you can see. That looks like the Foz de Iguazu, I guess, in the back, right? Something of that sort. So if you have any question, please. Do you any of you have any question? We can ask him now. Any question? Okay. All right, Michael, I'll be sending you questions through my WhatsApp, but thank you so much. Really appreciate it. Oh, you have it. David.
I really appreciated your time and your contact with the Argentinian is all a new relationship with NIH and really probably with other countries you have more connection. But here was an event with the support of the Argentinian Council of Ophthalmology and the main universities. So it's very important to us start to working together. So thank you very much for your time and your great presentation.
All right.
Thank you.
Thank you, Michael. I will see you soon. You're missing great food and great weather here.
I bet.
Yeah.
All right, so it's time to move on to our next speaker, who is also just amazing. Professor Salil Lachke. Professor Lachke is a distinguished alumni professor at the University of Delaware and has just been continuously funded by National Eye Institute for the last 15 years, which is, as you know, this is really very super task. So he's a super investigator in many respects, as what I would like to say. And he's the person who actually, professor who has the wet lab and dry lab, both experience that they have developed a system called EyeSight, which he's going to talk about. So without further ado, Professor Salil Lachke, please take it away. Thank you.
Thank you so much, John. Can you guys hear me?
Yes, we can.
Okay, fantastic. So because this is a 15-minute talk, I'm going to give you a quick sort of flavor of what we are up to here, developing these kind of tools. Okay. So very briefly, our research goals are to develop omics resources for predicting key genes in the eye. Basically, I'm going to focus on the lens because it has worked very well on the lens, but I presume this will work on the retina and cornea, as I would like to show at the very end of my presentation. And of course, once we predict these genes, we want to test whether these predictions hold true in human patients or animal genetics models and such. And eventually our goal is to be able to put together detailed gene regulatory networks, the relationships between regulators and targets, and then try to understand how the eye develops and of course what happens when the eye is pathogenic. So far we have identified several genes which are linked to human congenital cataracts or anophthalmia, microphthalmia, or Peters anomaly, and other defects too. The approach that we have used is also can be expanded to beyond the eye, and we have also collaborated with other folks to find new genes for orofacial clefts and tooth agenesis and so forth. But today I'm going to focus on the eye, and for now, I'm going to focus on the lens and the cataract sort of defects to show how this has worked superbly in that aspect. Okay, so of course, omics research is exploding right now. We have, of course, GWAS, genomics. We have proteomics, metabolomics, epigenomics, and transcriptomics. Okay, but all of this really informs us that it's no longer that hard to obtain large amount of data. And the key part is to be able to effectively mine this data to perceive key biologically relevant hypotheses that are going to get us the new genes and the new pathways to be able to understand disease mechanisms well. Okay, so the key challenge, I believe, of omics is to be able to effectively compile, analyze, and freely access this information, visualize this information in a user-friendly manner that really will be useful for the end sort of biologist or biomedical researchers or scientists or clinician researchers. Okay, and towards these goals, we have developed this system called as EyeSight. And EyeSight stands for Integrated Systems Tool for Eye Gene Discovery. It's a web-based resource, which I'll talk about in a couple of minutes. But before I do that, I want to say that all of this, although it's a web-based, omics-based research, is based on real biological data. This is, for example, I give this one quick example that, here is a well-dissected, very early E10.5 mouse lens, which has been marked by a PAC6 GFP reporter. And now, of course, we are doing single nucleus multi-omics, which I'll talk about in a few slides later too. But really, this is when you isolate a good amount of tissue and sort of well-dissected tissue, you can get good data out of this. Okay, but the key part is, again, how do you prioritize genes? Okay, and towards this, we have developed this system called as in silico subtraction. It's really based on concepts that genes which are enriched expressed in a particular tissue would potentially have a function in that tissue, and they are not necessarily very highly expressed. Okay, like for example, metabolic enzymes or such, they may not need to be very highly expressed, but they are exclusively expressed or more enriched expressed at a specific time point in that tissue, and therefore they may be important. That's the principle of this system. So therefore, what we call as subtraction is a way that we look at data sets with subtraction. As you can see, genes with, for example, housekeeping functions are completely removed, and genes with that function in the lens in this particular case, because these are lens data sets, you can see that they are highly enriched in this data set. So from the thousands of genes which are called as expressed in a particular data sets, you can home in on the select few genes that may be biologically relevant in that particular tissue. And over the years, you can see in the last 10 plus years, we have published several papers to keep updating this EyeSight website so that it can be used by researchers globally. Okay, so one quick example, if you had gone back and looked at congenital cataract linked genes in humans, which in their originally mapped intervals, which you can see are ranging from half an MB to like 46 MBs, which have got tens of genes, of hundreds of genes in there. If you use this system with EyeSight, you could have predicted the key genes within the first one or two sort of ranked genes in this particular system. Okay, so again, where do you go this? You go to this URL. This is of course published. You can even just Google ISYTE and Delaware, for example, or my name, and this site should pop up. And basically, it's very, very user-friendly for biologists or clinician scientists to use. You can query your gene as it can be, put here. You can select some parameters of which development or other sort of aspects you want to look at, and out pops this sort of data where this particular gene, for example, TDRD7 is expressed from 10.5 onwards, but you can see it gets enriched as the lens develops through developmental time. And of course, all the key genes, for example, these are crystalline genes. We know most of these are mutated in human patients with cataract, and they can be highly, easily sort of, visualized as well as, looked into, examined by this tool. Other genes which are not highly expressed can also be looked at. These are transcription factors. Again, these are also highly, easily identified by this tool. And of course, in addition to just expression in wild type tissues, and this particular data comes from the mouse, but we have also used knockout mouse models. So now again, at your fingertips, you can just put your gene in there and see whether that particular gene is downregulated or misregulated, upregulated in a particular mouse model with cataracts. Okay, and although right now eyesight is predominantly transcriptome based, we have also used proteome based data. Now we have started incorporating that, and here is a embryonic day 14.5 lens and retina proteome that is also included in eyesight. So over the years, this has been used by researchers over the world over. Many new genes have been discovered because of eyesight. Here are the human cases. We have also used it to add support to the GWAS predictions. This is a recent paper that I collaborated with Dr. Ellen Chokwe from Kaiser Permanente Institute. And of course, over the years, we have also found new genes and new pathways in animal models. So how does it work? If you have a mapped interval, or if you have an exome-seq where you have some candidates, you can go to the UCSC browser and you can see, for example, here, the most bright sort of red spot here is indicative of highly enriched expression. In this case, this is an example I'm giving of a gene called TDRD7. This is a Tudor domain family protein. This is generally important in spermatogenesis and germline development. So there is no way that we could have predicted that this was going to be important for eye or the lens if we didn't have this sort of an unbiased resource. And of course, eyesight predicted that this is the important gene in this particular patient which had congenital cataract. And very long story short, because this has been published 13 years ago, we have shown that mutation of TDRD7 in humans, or if it's a deficiency of this particular gene in animal models, gives rise to 100% penetrant cataract phenotypes. So of course, here is a quick couple of pictures here to show that, look, here is a gene beautifully expressed. This is TDRD7 expressed in the spermatogenesis pathway in mouse testes, that's on the left. And you can see a beautiful expression of TDRD7 in lens as well. Again, no way we could have gotten to this gene unless we have to use this unbiased resource. Okay, and of course, as this prediction of the expression shows, if you make a knockout mouse model, you get azoospermia, which is on the left, and a lens 100% penetrant cataract defect, which is shown on the right. And it's also, this particular syndrome that we predicted using a mouse model turned out to be, confirmed by researchers later, when they found a syndromic case in humans a few years later. Okay, but we discovered the gene and the first human pediatric and congenital cataract cases 13 years ago. Anyway, armed with this information, now we of course went with different new genes. For example, this is SELF1, another RNA binding protein. You can see it's conserved from fish to mammals, and you can see that if you knock it out or if you have deficiency of it, it gives rise to cataract phenotype. Another gene, RBM24, you knock it out and you get anophthalmia. It regulates SOX2, which is mutated in 40% cases of human anophthalmia, basically. And just in the last 13 years, we've been able to piece together a detailed post-transcriptional regulatory network for cataracts, lens and cataracts. And the reason I'm showing you this is because I want to show the power of this unbiased approach to for the discovery of new genes and new pathways in eye development. And of course, this is based on bulk RNA-seq, microarrays, which are kind of slightly older technology, although very effective technologies. But now, of course, we are expanding this eyesight database by using single nucleus multiomics. And by what I mean by single nucleus multiomics is that combined ATAC-seq, which gives you the chromatin status of a particular nucleus, and of course, RNA-seq from the same nucleus, which is a very powerful tool. And so, of course, here is, again, I'm showing you 3 different populations in the lens. They completely make sense with the number of the kind of genes that are expressed here. For example, you can see CDH1, which is ECAD, is expressed in the epithelium more than in the fibers. caprin 2 is more in the fibers than in the epithelium and so forth. You can also get, because it's ataxic, you can see, for example, how in DNA stupata you can see peaks which are potentially controlling the expression of this particular gene getting highly enriched as you go from the epithelial fate to the fiber fate. So of course, the important question is, can we use this eyesight approach to the retina? And we have looked at just the published retina or freely available retina data sets, which have not been analyzed in this sort of meta-analysis manner. And we have analyzed them, and you can see that, for example, with whole body subtraction, you can see the top list of genes, they're all enriched for eye and retina development. Okay. You can again go back and do a retrospective analysis, and you can see that you could easily find the genes which are linked to human, retinal defects, basically. And for example, here are many genes, which are well predicted. The more intense the blue color, the better prediction it is. And here are genes for retinitis pigmentosa or rod cone dystrophy or other sort of macular degeneration, for example. And of course, we can also expand this to cornea. We have done some preliminary analysis. Again, you can see, corneal genes, which are highly found by this whole body subtraction, in silico subtraction method. Here are again the same kind of retrospective analysis. You can find these genes which are linked to corneal defects very easily using this particular system. And so the take-home messages I really want to give here is that these kind of omics-based tools allow us to discover new genes linked to ocular development and their defects. But of course, they have to be also confirmed by animal models. And of course, the litmus test is to go back to the human cases and our GWAS analysis and such. Okay, but the price of all of this research is that you can get fundamental new insights into and new pathways into eye development and disease. Okay, so again, we have collaborators the world over, and we are forever looking for new collaborators of people who are interested in ophthalmic genetics to work with us and to try to extend this gene discovery model. And of course, I want to thank the NEI, without which support of which none of this could have been possible. So I thank you for your attention. And sorry, this is a very quick world tour for what we do. Okay, but if anybody has any questions, please feel free to contact me. Thank you so much.
Thank you very much, Professor Lachke. Let me just ask the audience if there are any questions here before we let you go. I know you have a busy day. You just got back from ICER, I believe, right?
Yes, I did.
Yes.
It was fun.
Yeah. Sorry, I saw you from a distance, actually, but by the time I got to that, you had already disappeared with your collaborators, actually. So hope you had a good trip.
No worries.
And yes. And hope you enjoyed the good food and good weather here. But any questions?
Love Buenos Aires. Yeah.
Yes. Okay. Any questions here in audience? You want to all think about it? I guess they are thinking about it. So
sure, no worries.
Yeah, I'll contact you if there are any questions here during the break. But thank you again. Take care, and after a long trip, you deserve some rest. So thank you.
Thank you so much. Thank you.
Bye-bye.
Take care. Bye-bye.
Okay, so I'm going to turn it to my co-chair, Professor Pelagius, here to introduce the next distinguished speaker, who is very distinguished.
Well, thank you, John. To close, the first section is about genetics, and after the break, we start the second session about artificial intelligence. The last speaker is my dear friend, Professor Natarajan from Mumbai, India. Professor Natarajan is a very well retina specialist and was a past president of All Indian Ophthalmological Society, Asia Pacific Trauma Society, and also is the General Secretary of Asia Genetic Consortium. And the topic will be bringing eye genomic research to the broader patient care and clinic. Thank you. Oh, I forgot this. Sorry, one small. And is the 76 countries, you know, so it's very unique.
Thank you. Is the microphone audible? Yeah, thank you, David. And my little brother, or younger brother from the past birth. So thank you, David, for all the love and affection. And we are friends for more than 2 decades. And thank you for giving me that fellowship called FELAS, Fellow of the Latin American Society. And also thank you for keeping me as the visiting professor of Maimonides University, Buenos Aires. So gracias, muchas. And greetings from India. So I'm happy to be here for giving this talk. Thanks to my friend, Dr. John Prakash, and also the director. This one? Okay. Okay. So my talk is on bringing eye genomics research to the broader patient care and clinic. And India and Argentina seems to be having similar, how do we? It's not moving? Oh, yeah, okay. Yes, yeah, right. So genomics, the eye genomics refers to the study of the genetic factors and variations that influence the development, progression, and treatment of the eye diseases. You heard from many of the speakers before, including Dr. Takeshi, who is our president of the Global Eye Genetics Consortium. It encompasses the analysis of genetic mutations, polymorphisms, and gene-environment interactions that contribute to both common and rare ocular conditions. And how genomics applies to ophthalmology, many eye diseases have a significant genetic component. And that's why Arnold Sorsby, about 60 years back when he wrote the book in 1947, he mentioned that ophthalmology is the playground of genetics. And I'm glad my father, late Dr. N. Sundaram, who started a genetic eye clinic in the government eye hospital in Madras in 1984, along with our colleague, Professor Kumar Manikvel. It encompasses the use of advanced genomic technologies to understand the genetic architecture underlying various eye conditions, ranging from common eye disorders like age-related macular degeneration to rare genetic diseases affecting vision. They're rare for the world, but in India, because of the ancient way of consuming as marriage, we have a lot of genetic eye disorders in India, maybe a few million. So the eye genomics research covers a wide spectrum of ocular conditions, including macular degeneration, glaucoma, inherited retinal diseases, and refractive errors. And in India, many institutions are working on inherited retinal diseases. And me being a vitreoretinal surgeon, my interest is in inherited retinal degeneration. So the early detection or detection of genetic markers before symptoms manifest, allowing for early intervention and management. And this is what we are doing in India. We are having a tough time to educate the people and the parents for this problem, because many treatments are in the research and many have not still come. Even the clinical trial have not reached India. I don't know about Argentina, whether you have the clinical trials going on. So evaluation of genetic risk factors to determine susceptibility and inform preventive measures, family counseling and genetic education, prediction of disease progression and severity based on genetic profiles, aiding in patient counseling and treatment planning. So next generation sequencing, genome-wide association studies and bioinformatics and data analysis, we need a lot of funds for that. And the recent advances in identification of novel disease genes like RPGR and CEP290 that cause retinal degenerative diseases, providing insights into disease mechanism and potential therapeutic targets, and gene therapy breakthroughs. Clinical trials and research efforts have demonstrated the feasibility and efficacy of gene replacement therapy such as Luxturna for RPE65 mutation in restoring vision in patients with certain genetic forms of blindness. The improved outcomes, cost effectiveness, improved diagnostic and early detection, family counseling and risk assessment, and patient empowerment and engagement. And integration with AI and machine learning is a future direction for the genetic research, ethical consideration, advancements in next generation sequencing. And from India and maybe from South American countries, we are looking for reducing the cost and also single-cell genomics and epigenomics. And the challenges in clinical interpretation, complexity of interpretation, integration into clinical workflows, this is what I'm working in India, cost and accessibility, privacy and data security, healthcare provider education, and patient education. So improved diagnostics, the early detection of eye diseases, precision diagnosis, personalized treatment plans, tailored therapeutic strategies, pharmacogenomics. And as far as of India and we are concerned, it's still the future, but we are hoping it will happen. Innovative therapeutic approaches, gene therapy, targeted therapies, patient-centered care, empowering patients, genetic counseling. And we need interdisciplinary collaboration. Our president mentioned that. The geneticists and ophthalmologists, bioinformaticians, ethicists and patient advocates, and government agencies and funding bodies. We have a NIH representation, and thanks for NIH for giving funding world over for the research funding and also the regulatory support. And I think India is still in the regulatory part for the genetic research, and patient and community engagement, patient-centered research, community outreach, global collaboration, and that's why the entire meeting is for, and knowledge sharing, international networks, and data harmonization. So this is in 2017, we started in my father was an ophthalmologist and grandfather too. So in my late father's name, I started a genetic eye clinic, Professor Dr. N. S. Sundaram Genetic Eye Clinic at Aditya Jyot, and it was inaugurated by a local professor, and that's why we are running a team with the genetic research from 2015 onwards, but 2017, we are doing the clinical work in the clinic. So this is a slide I borrowed from Dr. Subur Huang. You can see the NIH there with the retina image bank from American Society for Retina Specialists, and also Bascom Palmer and many others, where gene therapy and CRISPR-mediated gene editing for retinal diseases, the research is going on. And this is a project of inherited retinal degeneration at my hospital. We have a process flow of genetic testing and counseling, demographic collection, ophthalmic evaluation and possible clinical diagnosis, pre-test counseling, and targeted exome sequencing, where the patients are feeling that we are educating the people that since they cannot afford and they are from the lower middle class or middle class, they say, since there is no treatment, why should we do a test? So we are trying to write a grant and get a grant for doing the test free for the patient, post-genetic test counseling, and then re-diagnose with the clinical, the phenotype and genotype, and then keep the patients ready for a gene therapy for future. And we have our own research and vision in ophthalmology, which is main to do research within our clinic and the various other hospitals in Maharashtra, India. Not moving.
Choroidermia is a rare genetic disease in which photoreceptors in the retina and underlying vascular layer, known as a choroid, degenerate.
So this is a video from
Degeneration
YouTube.
of the retina and choroid begins during childhood and develops into blindness in old age. Choroidermia is caused by a defective CHM gene. In the gene therapy, a functional copy of the gene is inserted into a virus vector. The vector is then used to deliver the gene to the degenerating photoreceptors in the retina. Six of the 9 patients enrolled in the therapy trial have reported improved vision 6 months following treatment.
So these results are encouraging. And so the simple thing, this is the Luxturna, which is our first FDA approved, which says that you can inject into the subretinal space. It diagrammatically looks simple, but it's not simple. You need a vitreoretinal surgeon to do a vitreoretinal surgery like David, and go into the subretinal space, and there are lot of things are going on because technology has to improve, and there are successful results which they are reporting, and how you can inject and which area to inject.
The first 6 patients treated at the Oxford Eye Hospital were all able to see better in dim light.
So
In
this
2,
is
the
a
effects
surgery.
have been described as dramatic. The scientists first modified a harmless virus to carry a working copy of a human gene that sufferers lack. They then used a fine needle to reach the back of the eye, where they inject the virus behind the retina. The virus then infected light-sensitive cells, releasing the gene, which started producing protein that helped to preserve vision.
Rather than taking a pill or proteins or taking tablets, we're actually correcting the disease at the genetic level. In other words, genetically modifying the patients who have the problem to put the gene back that's missing. Obviously, we're very excited because potentially gene replacement is a way of stopping the disease from starting in the first place, and we could genetically modify people to prevent them from getting the disease and hopefully prevent them from losing their sight.
Around 1,000 people in Britain suffer from choroideremia, but the same technique could treat far more common genetic causes of blindness, including age-related macular degeneration. Doctors are so excited by the results published in the Lancet Medical Journal that they've already started another trial. The procedure could be approved for more widespread use in as little as 5 years. Thomas Moore, Sky News.
So this is the subretinal space which we have to do a microvitreous surgery to enter the subretinal space and inject the gene. And I think that we have still technical things for how much gene to. Okay, and this is the last video on gene therapy.
Let me see that.
Caroline and Cole Carper have been through more than your typical siblings.
I can't imagine it being any different than the two of them being together and going through this together. You know, they've had canes together, they've had to learn Braille together, they've gone to school for the blind and visually impaired together.
Early in their lives, they were both diagnosed with a rare genetic retinal disease called LCA, or Leber congenital amaurosis.
The doctor really said, there's no medical treatments at this time.
Both Caroline and Cole were expected eventually to go blind, but their parents found an experimental treatment, and 2 years ago, the kids enrolled together in a clinical trial. It was with Spark Therapeutics, a young biotech company formed out of research from the Children's Hospital of Philadelphia.
The idea is that we are taking a correct functional copy of a gene and adding it back into the cells that have dysfunctional or missing copy of that gene or blueprint.
It's called gene therapy, and it's relatively uncharted territory in medicine. The technology uses a modified virus to deliver a healthy copy of a gene to make up for one that causes disease. Scientists have been working on it for decades, but only now are the first gene therapies approaching the market. Sparks is among the most advanced. The healthy genes are delivered through eye surgery. Once they're in place, they're designed to do the work of a normally functioning gene, meaning patients only undergo treatment once.
What we're doing is different. We're actually adding the blueprint in, and the cell then knows how to do the last step, which is to make the protein or the enzyme that might be missing, and therefore that is what creates the potential for it to be a long-lasting effect.
In trials of 41 patients, the therapy is shown to improve both vision and light sensitivity with no major side effects. Caroline and Cole were no exception. Before their surgeries, both kids had trouble seeing in low light. Now, though their vision's not perfect, they say they're noticing big differences.
Good things are to come. I went outside when it was snowing, and I was like, oh, I can see the snowflakes. It was really cool, like, to actually see something that I've never seen in my life before.
Spark is one of several companies developing gene therapies, targeting everything from hemophilia to sickle cell disease to other rare disorders. If they're successful, it will be a new paradigm for medicine, fighting genetic disease in just one treatment.
I thought it was really cool how simple it was and how much it changed my life.
Hey CNBC fans, thanks for checking out our YouTube channel. Here you're going to find videos packed with all of the information that you need to be smart.
So my friend in Belgium, she's Professor Fanny. She's actually Indian born but settled in Ghent, Belgium, and she's working on the robotic retinal gene therapy. The reason is manually when you do, there is supposed to be some tremors, and this is supposed to remove the tremors, and you can see the robot part is used mainly only a part of the surgery. That's why there was a joke that the patient comes and says, and this is the gene thing which is being introduced into the robot, where this is done manually by the surgeon, and then the gene is, the robot is placed in the proper position. There are two elimination, which is eliminating the back of the eye, and then you keep the probe and then fix it up, and then the robot will take over. We can inject the exact amount in the subretinal space, and this is where the you can see the intraoperative OCT, and she's working with the 2 more companies on this, and I don't have any financial interest. My dream as a vitreoretinal surgeon is bring from the bench, from the lab to the bedside, and that's what thanks to NIH for giving grants to world over for doing this research. And I think there is a special issue on the journal, Human Gene Therapy. The eye presents a unique opportunity to study the delivery of therapeutic genetic material, because more than 300 genes are involved in the eye, in any of the genetic. So in conclusion, advances in gene editing technologies and gene therapies offer promising avenues for treating previously untreatable genetic eye diseases, and potentially restoring vision and improving quality of life for affected individuals. So it's our duty to explain to the patient that it cannot get a dramatic recovery, but as you saw in that 2 children who were done in the US, where they're able to, but they definitely improved their quality of life. And as we are essential for clinics, practitioners, researchers, and stakeholders to collaborate, innovate, and advocate for the responsible and ethical use of genomic technologies. And together, we can harness the power of genomics to revolutionize eye care, improve patient outcomes, and ultimately enhance vision health globally. So that's how, from India through US, thanks to Dr. David for having here, and I'll be happy to answer any questions or suggestions you have.
Because Argentina belongs to the third world, and for us it's not so easy. Argentina belongs to the third world, and for us it's not so easy to access to a treatment like that. How's the situation in your country?
Same problem, but that is why I'm friend with David to see how we can collaborate together and get these things. We have to educate the government. That's why, as a leadership development program, we are doing advocacy, because India, we still don't have anything, no clinical trial for gene therapy has started. And several institutions are working, and Dr. Kapil Parde from NEI is working with L.V. Prasad in Hyderabad in India, and Narayan Netralia is working, and we from Aditya Jyotai Hospital also are working. But still now, we have a lot of hurdles, but I think we have to cross the hurdles and get to our patients who cannot afford this treatment, and they cannot travel also. So I think similar situation in Argentina and in India. That's why we need to collaborate, and do something. And we have to impress, apart from the grants for doing research from NEI and our own governments, but even we have a lot of big business tycoons in India who are I know one of my company, he has a factory in Argentina, and I think he's also working with the Argentina, the commerce group. So I'm trying to impress him how we can do research. You can fund for the research and take the claims of supporting Indian people. And there are multiple companies like that who are having factories all over the world, and they live in India. So they still do only charity, but not on research, and then bring the research from lab to the patient. And that's where we are all here. Thanks to John Prakash and Director NEI for giving the opportunity for collaborative research. And a lot of my colleagues have got grants. I'm also trying to work on diabetic retinopathy and other things to prevent blindness. And inherited retinal degeneration is close to me, because I've seen as a kid my father working on it. That time, absolutely nothing in 1980s. And now we have 40 years later, we are still better off in knowledge, but we are not able to get to our patients. I think same problem in Argentina. We have to impress, apart from government, the rich companies who have a lot of money and who can give something for the patient care.
Just a quick comment here. As Dr. Natarajan said, there are a lot of things going on, and there are a lot of opportunities all over the world. But I have a really amazing opportunity here. An amazing opportunity that I get to see our eye research and applications and eye care and eye health all over the world. And I have the greatest honor and pleasure to work with Professor David Pelias kind of people from around the world. So all I can tell you at this point that we need, in order to get anything done in any country or any part of the world, whether Argentina, Latin America, anywhere, we need some champions, because you need to take the initiative, and then we'll all come around and support you. So without champion, it's impossible. But I'm so delighted that you have champions like Professor Pelias here. I'll turn it to him.
Thank you. Thank you. Yes, I am fully agree. We have a lot of people here working in the field, and for sure, with the Argentinian Council of Ophthalmology, we can start a project with the National Eye Institute and working together in the near future. Thank you, and Professor.
Now we have already finished the first session, so let's go to a little break. Just 15 minutes. Thanks.
Okay, so we tell the YouTube viewers that we have a break. How many minutes?
15 minutes.
15 you can. I think the camera is here. Okay, so the viewers can have a 15-minute break, and this part of the world you can have lunch, and the rest of the world maybe you don't go to sleep, have coffee, and come back in 15 minutes.
Okay, thank you. We start now the second session of the symposium, and in this second part will be about the different artificial intelligence applications in ophthalmology. We start with the first presentation. Is the progression of diabetic retinopathy a real-world data program? And it's a work we start to did in 42. And it's interesting because we make this program under the umbrella of the Argentinian Council of Ophthalmology. So as introduction, as you well know, in 2017, we have around 451 million of people with diabetics in the world, and increase around 693 million by 2045. And almost the half of the people with diabetes disease are no diagnosis. Also, the healthcare spend a lot of money in the treatment of diabetic population and is estimated in 850 billion in 2017. Between 40 and 45% of the diabetic patients at the present have some stage of diabetic retinopathy, and wherever less of the half of the patient of this condition are diagnosed at the moment. So as you know, we have different technology to make the diagnosis and different consults. Never in the first one, they need a second preparation to the fundus, make angiography, OCT, or different studies, of course, are also the fundus. So take time, the patient need to be at least 2 times in general. So start with different programs, as you can see in the slide, the program of artificial intelligence with application in the diabetic retinopathy. As you can see in Spanish, was in 2022, we make this open to all universities and group in Argentina to be part of this first experience in the real world of the application of the artificial intelligence in diabetic retinopathy, and we use the eye-art system. So we need in the first place, the people know how work the system. Then the other problem was we need a special camera to take the image. Unfortunately, this system works only with some special no mediatic or mediatic camera, not to all machines. So this was a problem here because was a limitation in the time we select the group of group of investigation participate in the program. And also, as you can see here, we have a group of 500 of pictures of the fundo image, and the result was compared with the retina specialist fundus and also with other instrument of diagnosis of the OCT, and in some cases angiography. And we will see what happens. Well, regarding the program we use, as you can see here, compare the 2 ways to make the diagnosis. The first one is in the Office of Ophthalmology. So take time. You need to make, the interrogation, the diagnosis, prepare the pupil to the fundus, and if the doctors saw some lesions, well, take it other studies. But when you use this program, only need take a picture, and in less of 20 seconds, you have the results. And the results could be the derivation to the retina specialist, the control in different times, or the patient don't have any lesion, as you can see here. And also, some cameras don't need dilatation. So, So you as you can see here, and also in between 20 seconds and 1 minute, you have, as you can see there, the report with the patient data, the lesion, and the recommendation of the artificial intelligence, what we do with the patient. As you can see here, different classification as no retinopathy, initial, moderate, severe, or proliferative retinopathy, and with the different diagnoses, you have a different recommendation. Well, was the number of the center. Was 9 centers with the distribution between Santa Fe province, Buenos Aires city, and Chubut center, 9 centers. So we collect this data. Well, at the beginning, this study was prepared in 2020, but for the quarantine, we start in the 2021 to collect the data. And all this center was opening. We not selected the center. The center sent and respond to the invitation of the Argentinian Council of Ophthalmology. So you can see here what happened with 500 cases. So the program don't having referable the diabetic disease in 66.15% of the cases. A patient need to see the retinologist 22.95%, and in only 0.9% of the cases, we have some problem with the obtain of the image, maybe because have cataract or vitreous hemorrhage or whatever. You can see here the results regarding the same data you see in the other slides, but in number, in the total of 500 of cases, we have patient don't need mediatric of dilatation of the pupil, 275, and with mediatric 225. Also here you can see the specificity and the sensibility of the cases, and also you can compare, and the system is worth more if you compare with the doctor. Well, one limitation of the study was the system need some clear medium to take the picture. As you can see here, with opacity, really the system don't work so well. And also in no-midriatic camera, only we have 2 cases from the 9. So unfortunately, it's a machine we don't have in too many clinics, on too many centers. And also the FDA have approved the iNUC system in some cameras, not in all cameras. So, and generally in Argentina, the people use the OCT camera as a fundus camera, so was a limitation in the study. So in this study, we can see what happened in the real world clinical practice with artificial intelligence program to detection of diabetic retinopathy. And the study demonstrates the value and easy to use this new tool, and can be used not only from ophthalmologists, also from endocrinologists, clinical diabetologists. So only need push the button and take the image, and the system inform not only if the patient have the diabetic retinopathy or not, inform if the diabetic need will see by ophthalmologists, and also if they need to see an ophthalmology very urgent because they have, for example, proliferative disease. So the system have the potential to reduce the cost, and is very easy to use, and also could be a very important role in the future to prevent the blindness in the diabetic retinopathy, also in very difficult geographic location from this patient, and also in area you don't have a retina specialist. So now we have a different groups publish different algorithms of artificial intelligence, and probably this model we can use with different machines. Now we have a lot of study use the OCT cameras and also cell phones camera. So probably in the future the patient have his auto evaluation, and the system say directly to the patient, please call to your ophthalmologist because your results are very bad. So probably when you organizing a campaign, you need take a camera, take a picture in very difficult weather or geographic or jungle or mountain or whatever, and probably in the future the patient only take a picture of the retina, and the patient know if needs to will see an ophthalmologist because retinopathy. Also we have program to glaucoma and macular degeneration. So well, this was the first experience in Argentina with artificial intelligence, finished in 2022. We really want to start 2 years early, but the pandemics we can do. So if you have any questions, if not, we move to our next speaker, Professor José Mendoza from Peru. He's from Lima, and they have the second presentation regarding other application of artificial intelligence. Thank you.
About the integration of artificial intelligence in an ophthalmological practice. Good morning. Thank you very much for the invitation to talk in this meeting. Today, I'm going to talk about the integration of artificial intelligence in an ophthalmological practice. These are my financial disclosures. So, artificial intelligence is used in medicine to improve healthcare outcomes, diagnosis accuracy, and treatment efficiency. The integration in ophthalmology offers exciting advances in diagnosis, treatment, and patient management. Ophthalmology is actually the second specialty in medicine to have more papers written in the last 2 years about artificial intelligence. AI ability to analyze large data sets and identify patterns makes it ideal for assisting ophthalmologists in areas where precision, early detection, and patient management are critical. We use a lot of images. We have a lot of different diagnostic tools that artificial intelligence could help us working with. In clinical research, also, it helps identify patterns and correlations in images, contributing to new treatments, protocols, drug discoveries, and identifying trends in disease progression. We are looking at some new imaging techniques like OCTA and foveal avascular zone for Alzheimer's disease, not only ophthalmological disease. We're trying to reach more systemic diseases with the scans taken on our eyes. And artificial intelligence plays a significant role on this. So how can it be beneficial in an ophthalmological practice? It could enhance diagnostic imaging, automating screening and triage, predictive analytics and personalized treatment plans, clinical workflow optimization, patients' education and engagement, as well as surgical assistance and robotics, and of course, some R&D. There's a lot of room to improve here. So enhancing diagnosis images, we use a lot of retinal images, we use visual fields, we use OCT, corneal mapping. All of these imaging techniques could be more useful and identify new biomarkers using artificial intelligence. For example, for retinal disease detection, like diabetic retinopathy, macular degeneration, and glaucoma, we could use artificial intelligence algorithms trained on detection of this disease. Actually, research shows that artificial intelligence-based systems offer high sensitivity and specificity in detecting retinal abnormalities, often compare or better than human experts. In OCT, it can help interpret OCT images faster, providing quick, accurate assessment of retinal and optic nerve pathologies. As well in corneal mapping, for example, screening for LASIK surgery, or try to improve the diagnostic in keratoconus. This will help a lot, of course, in busy clinics. Here are some examples of retinal detection based on fundus photographs, some other protocols and algorithms trained with OCT, and some with corneal mapping for assistance in keratoconus detection. Here is an algorithm running on OCT. You can see that it identifies some critical zones, subretinal detachment, RPE disruption, and then intraretinal fluid with all these kinds of images and of all these kinds of signs. The machine identifies and then could diagnose the pathology using machine learning techniques or deep learning techniques in this case. For automatic screening and triage on busy clinics and on populations that are really, really big, artificial intelligence could help identifying patients at risk and prioritizing cases that require urgent care. It also could enable early detection of progressive diseases, ensuring patients get timeline referrals to specialists, improving the outcomes. These systems in large-scale screenings for diseases like diabetic retinopathy will reduce the need for routine in-person examinations in low-risk cases, and this will reduce the strain on healthcare resources. This, again, could be really, really advantage in places like Latin America. So the company I'm working with right now has combined and merged with Microsoft to promote patient and healthcare provider participation in pre-screenings for system and neurological diseases. We want to go into OCT images, color fundus images, and not only diagnose using artificial intelligence different kinds of ophthalmic diseases, but also identifying with oculomics a lot of different systemic diseases, including neurological and cardiovascular diseases. We are calling this healthcare from the eye, and it's been launched during AAO last couple of weeks in Chicago. In cases of predictive analytics and personalized treatment plans, artificial intelligence could help us to detect progression and prediction in such kind of diseases like, let's say, dry AMD, when it turns to wet AMD, to predict this kind of pathology, predict the progression in glaucoma as well, or diabetic retinopathy. It could personalize and adjust treatments with access to patient charts and genetic data. It could help tailor personalized treatment plans, customized regimens for macular degeneration treatments, or laser-specific surgeries. The clinical workflow optimization in busy clinics and big hospitals is really, really important. AI can streamline administrative tasks such as scheduling, billing, documentations, allowing practitioners to spend more time with patients. It could also automatically take note-taking. I've been working with a clinic in Colombia. They're using this, and the workflow efficiency and the patient care, it's really awesome with this kind of things. Right now, we need to work We need to type in the chart. We need to spend a lot of time from a really small time that we have to diagnose patients, filling up a lot of charts and a lot of paperwork. If we just could dictate the machine and the machine will take notes and it will only separate the important words while we are discussing our clinical outcomes with the patient, that really improves a lot and significantly the workflow in the clinic. It could also help a lot with patient education and engagement. Taking an example, if you call an airline, you get a chatbot first. If you call the bank, you get the chatbot first. So this kind of chatbots and virtual assistants can provide 24/7 information to patients, helping them to understand condition, treatment options, and also pre and post care. These assistants can help educate patients about the right conditions, procedures, treatment options, and also engage tools to improve adherence to treatment plans. Personalized reminders can encourage patients to adhere to prescribed treatments, attend follow-ups, and manage chronic conditions more effectively. Also, surgical assistance and robotics is important nowadays. You have seen Elon Musk got a new robot, and also the Chinese companies, they got a lot of new family robots. We've got robotics in surgery, certainly for a while ago, like the Da Vinci robotic surgeon. This is a surgical robot that it's been produced by Sony, and you could see it's doing microsurgery right now. In surgeries such as cataract removal or retinal detachment, this kind of robotic assistance will be really, really helpful. Procedures like LASIK and other refractive surgeries, it could optimize the treatment, it could optimize the parameters in real time, and that will enhance the safety and accuracy that we have on our surgical procedures. Talking about Elon Musk, last week also, Neuralink has received the FDA's breakthrough device designation for brain chip implant. It's called Blindsight. It is aimed at restoring the vision. So they are implanting this Neuralink in the brain, and then with an outside camera or a functional outside camera, they're getting the signal. It's something like we've used in a lot of years ago for like the cochlear implants for hearing procedures. Right now, this company is aiming to blind people to restore their vision. And Neuralink is also testing another implant to aid paralyzed patients in using digital devices by thought alone. So this might be, we like a couple of years ago, we think of this like the future, but this is actually what is happening right now. So also in the space station, for taking an example of where the science and artificial intelligence is taking a lot of advantages, there is something that it's called SAMS. It's one of the largest barriers to astronaut health. It's an eye-centric condition associated with neuroocular syndrome. It represents a collection of neuro-ophthalmic findings observed in astronauts after long-duration space flight, including chorioretinal folds, posterior globe flattening, optic edema, and hyperopic refractive error shift. So imagine mankind is planning to go to Mars. That is a 6-month trip in outer space. How this condition could worsen and how it will make even difficult the way man becomes an outside of Earth, an interplanetary species. This can be really, really important to address this kind of things. And using artificial intelligence and OCT, we're getting a lot of analysis in the International Space Station for these astronauts to try to get better outcomes and try to facilitate mankind being an interplanetary species. There are a lot of challenges to consider, like data privacy. We must protect patient data and comply with all the relevant privacy standards. It is critical. We need regulatory compliance, ensuring that our IA tools meet regulatory requirements such as FDA approvals for diagnostic tools. That is also really important. acceptance and training. Ophthalmologists and staff need to be trained using artificial intelligence tools to maximize their potential benefits. We don't need to be afraid of these technologies. We do not need to feel like this technology is going to take our works. This is here to help us. We are going to actually being seeing more patients using this kind of artificial intelligence aids. The workflow will be more efficiency, and we will streamline healthcare healthcare in different countries. Sorry about that. So ethics, this is really important. There are a lot of countries we don't have regulatories about this. We need to have an important ethic committee to follow up some requirements. Actually, I think that I was talking with our friend David Pelaez here, and he said that there's a meeting coming soon that they will talk about all this to try to get the same kind of ethics and bias in the different kinds of artificial intelligence algorithms. So in summary, the integration of AI in an ophthalmological practice could help us driven treatment recommendations, drug response prediction, surgical planning and precision, automatic surgical skills assessments, clinical trial optimization, new drug target identification, remote retinal screening, wearable devices for continuous monitoring, for example, IOP with contact lenses, advanced imaging techniques and 3D modeling for seeing the whole picture of the eye, AI-powered retinal layer segmentation for better identifications of new biomarkers, AI-based predictive analytics for patient workflow. All this is important, and all this is happening right now. We are seeing just the tip of the iceberg. There is a lot of room to improve. There is a lot of work to be done, and we are so lucky to be living in this era of different changes that will lead us to this new kind of technology. I want to thank again for the invitation talking today, and I want to remind you guys that we have a Latin American Society of Imaging and Artificial Intelligence. We will have our 5th meeting. This will be an international meeting during EVRS meeting in Budapest between the 14th and 17th of November. We will have an hour meeting in Sunday, November 17th at 10:40 a.m. So please, if you are in Budapest and are coming here, please join us, and we will have some interesting discussion over there. Again, thank you very much for your attention and for letting me speak today here.
Okay, another amazing lecture. And okay, I'd like to introduce our next speaker this afternoon. And it's René Cano Hidalgo from Mexico. He is associate professor at the Universidad Autónoma de México, and he belongs to Fundación Balenciaga. And he is going to give us a lecture about biomarkers in macular hole and potential to AI. Thanks.
Thank you very much for inviting me to this important symposium. I am Dr. René Cano Hidalgo, retina specialist of the Instituto Conde de Valenciana at Mexico City. I am General Secretary of Eurolam Retina, and I am the President of SOLIA, Sociedad Oftalmológica Latinoamericana de Imágenes e Inteligencia Artificial. I will talk about swept source angiography OCT in macular hole and its relationship with artificial intelligence. My financial disclosure. As you know, swept source OCT is a very special technique that allows us to make a very special and profound exploration of the posterior pole. Swept OCT has a deeper penetration into choroid, vitreous, and sclera. Swept OCT angiography is an advanced non-invasive imaging modality that uses flow-based contrast without the need for intravenous dye injection to visualize the multilayered retinal circulation. Subsource OCT angiography allows us to have a large field of view facilitated by high imaging speed at 100,000 A lines per second, image fidelity enhancement with less motion artifacts, and potential for even faster OCT angiography imaging. A macular hole is a full-thickness defect of neurosensory layers of the retina involving the anatomic fovea of the eye. The OCT features of macular holes include a full-thickness defect in the neurosensory retina. There may be cysts in the neurosensory retina surrounding the area of the hole, as you can see here in this image. OCT features of macular holes includes a cuff of subretinal fluid that may be seen around the defect in the retina. The vitreous may be attached to the hole with vitreomacular traction, or they may be an operculum seen in the posterior vitreous on OCT scanning. Here is the operculum that is floating in front of the macular hole. The chronic macular holes has different features. In these cases, include chronic macular holes that show loss of the cuff of the subretinal fluid. There may be also retinal pigment epithelium atrophy seen in chronic holes that give us a reverse shadowing image. The localization of vessels, in particular retina and choroidal layers, as you can see here, the orange circles represent superficial retina vessel plexus. Green circles represent deep retina vessel plexus, and red circles represent choriocapillaris. But which aspects have been studied in OCT angiography in macular hole? We will talk about the description of morphological findings and artifacts in OCTH in macular holes. Different findings in OCT angiography depending on the stage of the macular hole. If we look at the subsource OCT angiography image of a small stage 2 macular hole, as presented in this figure, even if the structural SSOCT shows a full-thickness fovea defect, we do not see any pathology in the angiography SSOCT image. If we look at the superficial vascular plexus image, the vasculature is usually normal, and the size of the avascular zone corresponds to the size of macular hole. However, in a stage 3 macular hole, in this figure, when some tissue is present in the area of the full-thickness macular hole, the center of the fovea presents as a hyperreflective area, both in the anterior vascular plexus image and deep vascular plexus image. The deep vascular plexus image shows an increased avascular zone with a strange defect outside of the partially preserved vasculature. Close of the center of the fovea, we can just make out some vascular tissue but with a very low signal. Looking at the segmentation lines on the B scan, we can see that they move out of the filmed tissue or go through cystic space. In the center of the fovea, in the outer retina image, we notice the bright appearance of the choriocapillaris. The choriocapillaris in the center of the fovea presents higher reflectivity when compared to the rest of the layer, as you can see here. The increased avascular zone. Next, we presented a case of a small stage 4 macular hole. The superficial vascular plexus is once again normal, but we see an enlarged fovea avascular zone in the deep vascular plexus. Looking more carefully at the deep angiography image, there appears to be some vasculature close to the center of the fovea, but it is very pale and it is probably a result of false detection of the vessels, as the scan has been affected by the cystic spaces around the macular hole. Here is the increased avascular zone. In a stage 4 macular hole, sub-source OCT angiography without manual correction, where the segmentation has failed and the image present artifacts. If we have a stage 4 macular hole, especially large-sized holes, we see enormous superficial vascular plexus, but in the deep vascular plexus image we may see a hyperreflective area of vessels in the center of the fovea, where we know no vessels are present. This is caused by inaccurate segmentation, when the segmentation lines created automatically by the device may travel down under the retinal pigment epithelium and image choroidal vessels as if they were present in the center of the fovea. Such artifacts can be dealt with by manual correction of the shape of the segmentation line. The choriocapillaris layer is more clearly visible in the center of the fovea when compared to the other areas. In cases of full-thickness macular holes, this is probably due to the absence of retina tissue and vasculature in the center of the fovea, which do not produce any projection artifacts in the deep layers. We can see here the inaccurate segmentation, the angiography of the outer retina, and the angiography of the choriocapillaris. These types of imaging complications are even better illustrated in this case of a full-thickness macular hole with surrounding cystic spaces. In such cases, the delicate appearance of the vascular plexus may be presented around the cystic spaces. Initially, the image may suggest that some abnormalities of the vasculature are actually present. Here are the cystic spaces we talk about. Subsource optical coherence tomography angiography of full-thickness macular hole may be in case using manual artifact removal. The upper images present a macular hole in a superficial left and deep right retinal vessels. The oval hyperreflective shape in the middle is an artifact. The lower pictures were transformed with temporal and the flickering noise reduction algorithms and plugins in Adobe After Effects in order to remove the central artifact. We noted areas of decreased perfusion on the upper margin of the macular hole. These probably correspond to retinal edema around the macular hole. Here is the manual artifact removal. And what about the vascular features in OCT in macular hole? Spectral domain optical coherence tomography via scan of a full-thickness macular hole we are seeing in this image. A manual outline of retinal tears indicate the location of the perifoveal cystoid cavities in the inner nuclear layer in blue, and in the complex consisting of the outer plexiform layer and Hendel fiber layers in red. Here are the perifoveal cystoid cavities in the inner nuclear layer. And here are at the outer plexiform layer and Hendel fiber layers. The retinal vascular differences between the healthy and pathological eye corresponding mainly to the deep network of vessels. Small, circular, hyperreflective spaces are present surrounding the macular hole in the inner nuclear layer, whereas elongated radial hyperreflective cavities forming a stellar pattern are observed in the outer plexiform Hendel fiber layers complex. Here We can see the retinal vascular differences between the healthy and pathologic eye, corresponding mainly to the deep network of vessels. About the evaluation of choriocapillary circulation. Some studies have shown that the anatomic alteration of choroids may be associated with the development of retinal defects and the progression of macular holes. Recent studies have found that the choroidal thickness was reduced in eyes with macular hole when compared with unaffected fellow eyes and healthy control eyes. There is still some controversy over whether the choroidal thickness alteration is a leading factor in macular hole formation. To investigate the choriocapillary circulation in the macular area for eyes with unilateral macular hole before and after vitrectomy using OCTA, this paper analyzed 25 patients. Choriocapillary circulation was measured by OCTA to obtain two measurements, flow area and parafoveal vessel density. The results are flow area and parafoveal vessels density of choriocapillaries in the macular area were significantly smaller and lower in idiopathic macular hole eyes than on unaffected fellow eyes and healthy control eyes. The choriocapillary circulation was negatively correlated with macular hole diameter, as you can see at these schemes. And about the evaluation of vascular and morphological changes following surgery is a very important issue. Characteristics of the foveal structure and retinal microvasculature in eyes with macular hole before and after surgery. Evaluated the relationship between vascular change in OCTA and visual outcomes PCVA postoperatively. To describe morphological of retina and choroidal vessels in swept source OCT angiography before and after vitrectomy with a temporal inverted ILM flap technique for full thickness macular hole. This is the paper and the evaluation of OCT features in patients with macular hole before and after vitrectomy. It's a prospective case series study of 28 eyes. Surgical closed macular hole eyes showed reduction of vascular zone, foveal avascular zone, an increase of foveal vessel density and choriocapillary flow and compared with these parameters preoperatively. In this other paper, it's a prospective observational study of 36 eyes using swept source OCT was performed in patients before and 1 month after vitrectomy with a temporal inverted ILM flap technique. The study demonstrated lower density of vessels in the deep retinal plexus in the area where ILM was peeled as compared to the rest of the fovea. In this paper, the purpose was to measure the foveal avascular zone area after ILM peeling. It's a retrospective, observational, and cross-sectional study. 102 patients with unilateral macular diseases and 169 healthy subjects. The vascular zone of the fovea area after internal limited membrane peeling was smaller than of the controls. A smaller area was correlated with a thicker fovea, both in internal limited membrane peeled eyes and in normal eyes. In this paper, they analyzed to determine the size of superficial vascular zone by OCT before and after surgery for an idiopathic macular hole. Is a retrospective observational case series of 16 eyes. Transplanar vitrectomy was performed with internal limiting membrane peeling. The significant decrease in this superficial phase of the macular hole surgery indicates that there was a centripetal movement of the foveal tissue postoperatively. In summary, SSOCT angiography analysis macular hole and the assessment of vascular involvement should be an important tool to identify change in microcirculation after vitreoretinal surgery. quantification of choriocapillary circulation might serve as a parameter to evaluate the progression and prognosis of macular disorders. The removal of ILM might be related to the postoperative reduction of foveal avascular zone size. The application of artificial intelligence can be very useful to know prognostic factors and surgical decision in cases of macular hole. Finally, I want to make an invitation to be a member of SOLIA, Sociedad Oftalmológica Latinoamericana de Imágenes e Inteligencia Artificial. We can contact us at this web page, and thank you very much for your attention.
Thank you, Professor Cano. And we move to our last speaker. It's a great pleasure to make the presentation of my co-coordinator, Amin, from Canada. As I made the presentation on the first time, Amin is a combination between the academic and the industry, and also made different companies working in artificial intelligence in application, not only in ophthalmology, in all medicine. So thank you, Amin, to come in. And I hope also probably the next year we start the first postgraduate course in artificial intelligence in ophthalmology, and Amin join us in the course. So thank you, Amin. And yes. Okay. We have an extra talk from Professor Natarajan. The title of the presentation is an Artificial Intelligence and Telemedicine Update. Amin.
Thank you, everyone. I think this is exciting to recap what Dr. Mendoza, our colleagues, have spoken about and how to actually bring it all together. That way. I really don't have any disclosures for this talk. What's important for me to explain again is there's a lot of excitement in the industry, but this isn't new. AI isn't something that just suddenly sparked upon us and suddenly we're trying to play catch up and the fever of excitement. But if you take a look at major milestones, I think when we look at 2015 with telehealth coming onto the picture and 2018 with IDX really being approved by the FDA are real milestones in retinal and ophthalmological work with AI. There we go. A word of caution, actually, before we start our talk. We get really excited about AI and its projects and where they are. But I think as we have other viewers from different parts of the continent, where you are in your programs and where we are really makes a difference. Are we still in the excitement phase? Are we have we hit our expectations? Like we have many of what we call screen AI solutions. Really, we've kind of peaked and really we're coming through in understanding. I wouldn't call it so much as disillusionment, but really realization of how important they really are. And of course, hopefully we can get to a steady state scenario. This is from Gardner, but I really caution all of us investors and project managers in terms of where is AI going to be. Why is there the hype? Not only in ophthalmology, but in healthcare in general. It's all about timing, I think, and the three things culminating together. We've got the advancements, of course, in machine learning and algorithms. We have enterprise solutions, in fact, now, where algorithms and machine learning are actually probably going to be commoditized themselves. You have Microsoft coming out with wonderful off-the-shelf algorithms that researchers can begin with and make their own. So the idea of course, natural language, LLMs coming together, and then finally, well, not finally, but of course, cloud computing, the compute power being available so we can share information and data securely. But I think the real key here is the availability of the data. In the end, it's really going to be about the data. What's the quality of the data that we're using? And of course, what data is the algorithm or the program really going to be built on? Everything else, quite frankly, is going to be a commodity. This is a little bit of for thought for us all who are viewing. You know, where do we stand? These are very well-renowned colleagues of ours that have some really strong statements. And, you know, it really depends on where you are and how you think. You know, probably I personally, I mean, again, I'm probably on the second half of this, the two comments, where I believe AI is a tool to enhance what we do. But I do believe very strongly that you know, it's in our own best interest and our patients to understand AI and how to implement it in our clinical practice. I you know, had a lot of discussions, and you'll hear them from you know, very renowned colleagues. And I kind of think it's very interesting, and I said it this morning on a sidebar, that the day may come really close where, you know, if we don't reference an AI in diagnosing something severely, that medically legally, could you actually be responsible? Just again, a quick one. I won't get into the details, but you know, how quickly has AI and models really been advancing? This is all that we're showing from medical questions being answered in 2022 in March to 23. You'll see just how incredible fast the accuracy has moved. And of course, in terms of the parameters and the sizes of these LLMs, how large and of course, in terms of their size becomes their power. But how quickly this is actually moving. And we've, you know, again, speaking of data, I think it really is about the data and the ability of having these open sets of data so that researchers aren't just financially burdened and can't actually build, you know, for their own regions and domain-specific LLMs. But I think that's what's really been helpful. You'll have these large data sets like the Kaggle and UK Biobank, which have been paramount in starting many different research projects. I think Dr. Mendoza talked about this a little bit, and I totally agree that, you know, with the as much as the positives come concerns, that responsibilities as both practitioners and policymakers really need to spend time on. And, you know, with interoperability, privacy, governance issues are really there. I think as clinicians, I always bring the idea of, you know, uninteroperability. You know, we're taught to understand how to make a diagnosis. And I think there's a mental block for clinicians to use this AI, this product that somehow spits an answer, and we really don't know how it does it. And as we get into overcoming that, it will, I think, make a big change in adoptability. But of course, the rest of them, we talked about hallucinations and bias, which are exceptionally, you know, real. They're real. And, you know, there's something that clinicians really need to pay attention to and retest these models. You know, where is AI today? You know, the rest of my talk is really a landscape on where we are. And probably the most effective use of AI to date in, you know, out in clinical practice are in, you know, the administrative world, which is terribly burdened and is just as important in our for ophthalmology and eye care. And you'll see that in education. You'll see it in all these referral managements. You know, claims processing, RCM solutions in the United States have been taking off in AI. And you'll see the solutions in scheduling, note-taking, like I think we've used Nuance and Dragon AI-based solutions. These are real practical applications of Gen AI. And as I alluded to, education, a huge opportunity for AI in medicine as a whole, in training clinicians, surgeons, in being able to, modify clinical workflows. And this whole concept of spatial computing, of AI coming together with VR and AR, to be able to help us educate and train as resources become more and more strained is going to be an incredible opportunity for us. And we alluded to this earlier in talks, but this is the sexy part, the stuff that we get excited, and that's what I was trying to say. The administrative opportunity is a huge financial burden, is extremely valuable. We spend a lot of time on this aspect, however, which is because we're clinicians, we like to do this. However, again, when we look back at oculomics, we had a talk earlier about that, but the biomarkers that are out there in the eye, it's absolutely incredible. So the eye, other than radiology, is frankly why there's so much excitement and hype in healthcare with AI, because we're so heavy on data and multi-modal type data, from text, EMR data, structured, unstructured, through visual fields and OCTs, images from anterior segment. So you'll see all of these projects that are out happening just for the eye. David, you talked about your project here in the real world. Of course, diabetic retinopathy was the first, one of the first, but it's so important that it's kind of a good learning option. And of course, that will now move to other modalities of diseases in the eye. But again, it's really about looking at, all over the world, how we've jumped on this, and everybody's taken it in a different manner, creating the same solution and AI solutions that they've all created, and learning from each other and how they differ. I mean, it's understanding the differences, performance metrics. There we go. And I think the holy grail that we're all trying to look for is a co-pilot. And I think this is going to take it's going to be quicker than we thought, but it's not magic. That, how will we be able to, I think I've missed a slide in between here. Yeah, I wanted to talk about this, thank you. Other than the diabetic kind of disease process, but where AI is, of course, creating a lot of excitement is in drug development, the ability for us to, and it's real partnerships that are happening. They're saving, if you talk to them, years of drug development that we couldn't actually go and search these data sets. And there's real partnerships with AI that are happening in industry today. The last, of course, the whole concept of personalized medicine. There is no way without, I think, AI that we could actually bring all of the data of our SDOH data, our EMR data, our imaging data, to really create personal care and work plans for our patients. And what's next? And I think Dr. Mendoza alluded to this, but the eye really is the opportunity we have, both financially and to create equity and access to care like never before through just one image. And I think, David, you said it too, with one quick photograph, how much can we learn? Not just about the diabetic retinopathy, which is a great use case, but taking that further into chronic disease, kidney disease, cardiovascular care disease, and the other ones. But I think the oculomics is really a burgeoning opportunity with AI that we really need to spend time on. And as a, we've all said a key to our soul through the eyes, but really it may be the key opportunity for healthcare in general. So as neurodegenerative diseases, these are real projects that are being worked on, that are coming to not only pilot projects, but we should see commercialization soon enough. And really excited to be able to do that and bring this sort of access to care through AI, through what we do each and every day, through OCTs and retinal imaging, and being able to diagnose, screen early to prevent downstream expenses in the healthcare system and our resources as a whole. So you're seeing work in Alzheimer's, Parkinson's, MS. I mean, it's quite incredible to see the speed that we're working on. So, it is really the future really is here. And you're seeing, we saw an example of a video of a robotic surgery through a Da Vinci system, potentially, holographic, imagine, David, you being able to not only screen the diabetic retinopathy, but then actually counseling a patient about that, almost as if you were in front of them. This is kind of the type of, and then personalized work plans. Imagine how our patient care in the future will look like. When we start implementing this is kind of some of the work that I speak about, is actually not only taking it from the clinic and from pilot projects, but how do we actually get it out, not only commercially, but into the hands of a patient. And I'm going to share with you two projects that that I've been involved in. This one was my own, a tele-eye care program. This was early days in Canada, where we used, I know in Latin America there are no optometrists, but again, similar concepts, but using all the resources that are available to prevent the travel and allow rural care. So very similar to the project, David, that you did. This was done, but we did this in, I don't even remember, 2019, 2018, something like that, to really create the outcomes and measure those KPIs of travel and saving dollars and cents. A similar project at a larger scale that I'm involved in with an organization in India, the LV Prasad Eye Institute, and they've been, again, doing a very similar project, but at a much greater scale. And an incredible opportunity for vision centers, referrals, large landscapes, rural and tertiary centers, very similar to what was described here in the landscape, and being able to again show validity, preventing people from travel, and increasing the access to care where it's needed the most. And again, being able to not only take telehealth to that, but you add the AI component to this and the triage, and now we have really effective use of technology and resources. I created a telehealth, I ran a telehealth company in Canada. What I foresee next is what I'll call telehealth 2.0. What does the future look like? It's actually, so telehealth is just distributing that technology, medical care in the hands from a distance, but how can now all these different technologies accumulating and culminating together is really the exciting part. When you can take the AI development we talked about in systemic care, not only eye care, but then now being able to distribute that into your homes and create longitudinal work plans and care flow, that really gets exciting and is really what's going to change over the next years. As a summary, here's what I see. We created an integrated platform in telehealth for general medicine. I see for eye care very similar. I see just a continuity of care. It's not a division between one technologies and others, but as we put them together, and this concept of multimodal medicine, that personalized care is really in the hands of patients, in the hands of their mobile phones. And that really is going to change the delivery of healthcare. Please, if you have any questions, my email is up there. I'd be delighted to answer and continue the discussion. Thank you for having me.
Okay, thank you, Amit. For your great presentation, you can see more what possible happen in the future, maybe more. So, it's the end of the symposium. We have a last presentation from Professor Natarajan, and thank you to all the coordinators, Amit, John, Nati, and Pablo, and coming.
Thank you, David. So this is in continuation. this is in continuation of the talk what you gave. So I made a slide, blind-free Argentina, my vision from screening to surgery for proliferative diabetic retinopathy. Because I wrote about 5 years back in a long flight that what's my dream for a blind-free world. And then I divided into the India, then my state, my city, and various states I'm involved. So I'm still in the, probably like the hare and the tortoise, and I'm still like the tortoise going slow. But because we all know that diabetes is the epidemic of the 21st century, and diabetic retinopathy is a major cause of blindness in the working middle-aged adult population, 5th cause of blindness globally. And so we saw this statistics like 2015 to 2040, it's going to be 642 million. And India has, as of today, 100 million diabetics. And then this was a few years back in 2017. And then, so this is what they are saying, it's going to keep on increasing. So it's an epidemic. Every diabetic is affected. It's an incurable disease, which is what I want to convey to the public. And eye symptoms are too late. So people are waiting for symptoms to go to an eye doctor. So prevention is better than eye care. So this is what we learned in the preventive and social medicine as an undergraduate some 4 decades back or 5 decades back. We never followed that, but now I think it's very important, even though I'm an aggressive retinal surgeon and I love surgery, but prevention is better than cure. That's what I come to know, because so many millions are suffering. Why they should suffer? Because of lack of awareness and lack of screening programs all over the world. Maybe the first world has some, but I know even in the United States, 50% of diabetics are diagnosed late because they are in the remote possible places and they don't have insurance and they don't go to the doctor. So effective screening program for diabetic retinopathy and determine who needs to be referred to an ophthalmologist for close follow-up and possible treatment. And this is the same problem, maybe a big country like Brazil and Argentina or all the South American country, maybe the physicians and the ophthalmologists are in the bigger towns and not in the smaller places, in the villages. So I made this as a president of All India Ophthalmic. We only have 1.4 billion people as a population, maybe 1/4 of the world, and every citizen deserves an eye checkup. So how do we do that? And so I started a foundation which is running for more than 2 decades in the slums of Mumbai. We thought before going to the villages of India, we have about 645,000 villages in India, and before going there, a lot of NGOs, government are working, but we thought there are municipal hospitals which is run by the government in slums of Mumbai, but still people don't go because they leave the house early morning for their bread butter and then come back in the night. So by the time the clinics are closed, so we go door to door and screen them. And these are the various places where we take our van or a motorbike. We did the first transferring the fundus camera, which is seen in the lower part, called the Forest, and I don't have any financial interest, which is made in Bangalore in India. And like this, we had satellite clinics, and wherever there were optical outlets, we did a screening program, and that is before the AI. And we also, our social workers go door to door and do a diabetic test above the age of 40. And no, because as I said, the recent publication in Lancet says out of 100 million diabetics, 58 million do not know they have diabetes, and that's a problem of India. I don't know about Argentina. So we are now going to every slum house, and we have covered about 300,000 slum houses. Where we go and enumerate how many people are there, and we have touched about a million people. But we go and find out how many are above the age of 40 and what is the family, and then whoever is above 40, we do a random sugar like this, and then if it is more than 150, we may then do the HbA1c. Everything is done free, but somebody has to pay for it. So I run this NGO in my parents' name called Kamala Sundaram Foundation, and our John Prakash gave the first public endowment lecture and talked about the research and what is going on by NIH and others to the public in India. So this is a World Diabetes Day. We do camps all over, and that's what I see we should do. This is a Zeiss camera given by the World Diabetic Foundation from Denmark, where we have a van, and we have the Zeiss camera, Zeiss laser, which can go. But in slums, the van cannot go because the van is very big. So we take it by motorbike. So we do camps in all parts of Maharashtra. So I think David mentioned that Argentine Council of Ophthalmology did the first diabetic retinopathy screening, and which is the largest. Same thing, we created a Guinness World Record of examining 649 diabetics. Both I funded the photograph, and we collected about 2,000 diabetics, gave them diabetic diet by another NGO called Roti Bank, where we serve daily 15,000 people free meals for the poor people in India, in Mumbai. And then, so under the National Program for Control of Blindness, Government of India is doing a great job in the center, and state governments also follow. And then they have employed ASHA workers who are females who work in the villages and all the public health centers. But the NCD, the non-communicable disease diabetes, is looked after, but not the diabetic retinopathy. So we're now trying to, the government is giving some reimbursement for if you screen a diabetic retinopathy, but the problem is the doctors are not aware, and then they feel, I also feel as an ophthalmologist, why should an ophthalmologist go to do screening? We can deploy anybody. That's why I made a program called Anybody Can Screen for Diabetic Retinopathy. So we create a pre-camp awareness assembly of the diabetics, which is a challenge because people don't know whether they are diabetic or not, and capturing of the images and diagnosis done by ophthalmologists, AI, or image graders. And here we are going to talk about finally using the AI. As David mentioned, nowadays there are a lot of companies having an adapter for an Android and iPhone, and we found there are several funders on phone available. And again, I don't have any financial interest. I work with the Remedio, which is made in Bangalore, India. They have a FDA approval and CE approval in Europe, and they're using a software which this is the fundus on photograph taken on the Zeiss photograph on the left and on the right, and the left is the you have the fundus on phone, and almost the photograph quality is good, so you can take a good photograph with the iPhone. And then we have applied the first offline AI in the world, where you can see the blue color which differentiates AI in the mild proliferative, non-proliferative with the CSME, and severe non-proliferative with CSME, and proliferative diabetic retinopathy CSME. We made two categories, even though it can classify everything. Refer to an eye doctor, and no need to refer. That idea is anybody can go to the villages or slum. That means not a doctor, not a technician. So anybody with the school dropout, we have trained people, and we make sure that they can go and use this camera. So there's a link between medically underserved area and tertiary healthcare services, something like that. Argentina should look for, so that you have an automated uploading of images at the central server. And the main thing is offline AI. We don't need the internet, and we don't need electricity, because the images are in the mobile phone, and the phone can be charged, and the infrared device also can be charged, and that's the first of its kind in the world. And the sensitivity and specificity of AI in detecting referable diabetic retinopathy was 100% and 88.4% respectively, compared to ophthalmologist grading, no false negative. And the sensitivity and specificity of the offline AI in detecting any diabetic retinopathy was 85.2% and 92% respectively, compared to ophthalmologist grading. So we also studied comorbidity along with the endocrinologist, and published this diamond study called the Diabetic Retinopathy Awareness and Association with Multiple Comorbidities. And I think the problem is, I keep telling every patient who comes to me in my clinic, and I teach my social workers who go door to door, that diabetes affects from head to foot. And if you diagnose eye and you can make him a healthy person and see well, I think he'll be a good thing for the economy, for the family, for the city and the country. So early detection and treatment after screening can reduce the prevalence of diabetic retinopathy even in the remote areas in any country, and a reduction in the economic burden among poor and illiterate people. Opportunity for training anybody who's a non-skilled worker, give him this training and make him a semi-skilled worker, and it'll help improve overall health of the population. and this I read about AI, the way forward. The next 3 decades will be the most technologically disruptive era in human history. Advances in AI, combined with radical breakthroughs in hardware, will usher in an era that sounds like science fiction today. Devices will fade away, virtual environments will emerge, and super smart AI assistants will organize our lives and run our businesses. In this new world, our relationship with technology will change forever. We will both virtually and biologically merge together. Even though we are doing this meeting online, myself, John, and Amit have come all the way to meet David in person, and we love to meet people apart from being virtually present. And I'm happy this is there in the YouTube, so it'll be recorded and it'll be there. So offline AI, we capture the image, analysis of the image is done in the mobile itself, and it takes only 10 seconds for the image to be studied and the results come. So there are IDX system, the first one which was approved, TopCon, which is expensive for India in US. And then you have, this is the IDX, which is there in the eye diagnosis. And then Wisconsin FPRC study on AI. And then Forest Health Cloud-based teleophthalmology software. Google's initiative, Google's DeepMind, working to bring AI devices to the market for detection of eye diseases. They're having hurdles on that, and even Microsoft and Google, they mentioned this, but at present I think I don't hear anything. 2019, Sundar Pichai announced that Google AI can predict heart problems by taking picture of your eye. And now I know Google Pictures say that the fundus belongs to a female or a male, but I don't know what's the use, but I agree there is a technology which is used. And I think the key benefits of AI, diabetic retinopathy, is a social and economic problem for any country, and early detection enables specialists to focus on the treatment, so that doctors like us, where we are a surgeon, we can treat these patients early, prevent the problem, but in case few of them develop complication, we can tackle them early and give them good results. And deep learning techniques, continuously improving outcomes, relatively low cost in India, and highly scalable process with quick response time. And so we are extending this project to the government, to the state, Rotary, Lions, Indian Medical Association, Association of Physicians of India, and anybody, I mean, anybody we want to. So we have this religious congregation of about a few million people, and this happened in 2019. We used the Uttar Pradesh state, and then did this camp for the people who are willing to come for screening. The idea was to create awareness. So I created this slogan, anybody can screen for diabetic retinopathy. That means you don't need doctor, you don't need technician, we need an offline AI so that anybody can be trained and then they can detect it, and the offline person cannot do anything except referring the patient. So the only out of 100 patients, when they screen in a day, maybe only 3 or 4 may need treatment. Somebody will need injection, somebody will need laser, and somebody will need surgery. So if they detect early, I think you can prevent all complication, and most of the patients can have 20/20 vision. So we had this research question in 2017, is AI reliable to pick out those in need for seeing an eye doctor? So that was the research question, and we published the answer in JAMA Ophthalmology in 2019. Thanks to my co-authors, diagnostic accuracy of community-based diabetic retinopathy screening with an offline AI system on a smartphone. And there was a commentary given by Alvin Liu from Johns Hopkins, and I think there are several presentations in American Academy saying this should be not the future, it already should be there, where they called it a future of ophthalmology in one of the symposium. And I'm happy to say from 2019, we are doing it regularly in India. So this, you see, the sensitivity is 100%. And then this was in 2019, the most viewed article. We are proud to say that a simple instrument can prevent blindness in the remotest part of any country. So the reliability of AI for screening population of diabetic retinopathy in the need of ophthalmic checkup by an eye doctor is 100%, and that the results are promising. So this is a video where you can see that anybody can be trained to use this mobile phone, which is attached to an infrared device made by the Remedio, and you take beautiful pictures. And here you see the person is putting the AI, offline AI, and then it will say that the photographs are 99% good. If it is good, then it will analyze and say there is retinopathy, or there is like this blue color uses the changes in the fundus and then sends to the, says the patient should be referred. And only these patients need to come to the clinic. And according to us, India has the maximum users of smartphone in the world. And I think almost 1 billion people use a smartphone. So the idea was to teach them to use the smartphone to screen diabetic retinopathy. So the advantage of using the offline AI, more job opportunities for and more working hands, we have skill development and early referral of referable diabetic retinopathy. And then you create awareness about diabetes, because a lot of people don't know that diabetes can produce blindness and the importance of regular follow-up. And then if they come to eye doctor, they can do the treatment, and probably 99 people or 95 people who don't need treatment can go back and don't have to come to a clinic, spend that much economy is saved for the country. So thanks to our former President of India who gave the name for my foundation, which is now changed to my parents' name. He came, and that's my best friend Shivanandan, who was the chairman that time. So we showed him how we do that. He asked me if using offline AI, will the examiner disturb or injure the eye? I said, no, sir, we don't touch the eye at all. So it's like a non-mydriatic camera, and we can use the phone as a camera and do it, and we can do. And then we also inaugurated, you can see my, that time, 100-year-old grandmother who was inaugurated this motorbike in 2014, where we carry the fundus camera in motorbike, in the scooter, in the can be, and we didn't try and cycle, but in the van where we can take to hook and corner. So the main thing is the whole idea of my talk is that we see me and probably David and Professor Cano who spoke, we are all vitreoretinal surgeons who are doing surgery daily to prevent these sort of sight-threatening traction retinal detachment, and these sort of patients with a non-clearing vitreous hemorrhage and then a combined traction and regumatogenous retinal detachment, taut posterior highlight. All these are because of uncontrolled diabetes and not examined the eye, rubeosis iridis producing vitreous hemorrhage and glaucoma, and finally blindness. So the answer is vitrectomy, and here I'm just saying that this is an expensive surgery where we have to go into a keyhole surgery. This is for the researchers to see that these sort of surgeries, each patient, the time and the money spent is so much, and then we could get good results. But the idea was to say that why not we prevent it? I mean, we see patients like this, and I've done a lot of live surgeries in Mexico. We see complicated traction retinal detachment, and then we have to do this advanced technology and spend 2 hours, 3 hours to repair the diabetic retinopathy. So all this time can be saved. So people, particularly the middle income group who has maybe the breadwinner, and if he has blindness in both eyes like this, and we have to operate, and who's going to put the money? Here, few patients we are able to do through our foundation, and government is doing through their government medical colleges and hospital, but still we have so many patients who cannot have access. So the idea is to screen so that you don't have to do this sort of expensive surgeries, and then we could get an excellent result. You can see the optic nerve is pale, but the vision was almost 20/80 by 6/9 vision. But I think we have to spend a lot of, and we also do now the latest technology of 3D surgery. And I think the idea is, all this will benefit the surgery and also the surgeon's ego. But our idea is to prevent these patients to go in for surgery, because in spite of doing all the prevention, 90% may be saved, and 10% will have to come to the surgeon for complicated vitreoretinal surgery, which is like this, you keep using the 3D. So I made another Section 8 company like the 501 in India, in US, to make India blind free. We want to make this a not-for-profit organization, which we have the 2 directors, and then from the future plan is we had seen a similar study done by Shobha Shivaprasad from Moorfields, London, where she impressed the government of Kerala and used the Remedio. Again, they started with 4,000 patients in 60 family health centers, and then this is what they did. They went through the Kerala non-communicable disease registry, 16 family health centers, 80 family health staff were trained by the Remedio, so that the government on their staff were doing this screening. 4,527 patients enrolled, and retinal imaging to Remedio, and the same thing can be transferred to the center. And you can see patient was screened for proliferative diabetic retinopathy, and if there was no diabetic retinopathy, follow up 1 year later, so they don't have to go to the ophthalmologist. Mild also 1 year, and then only if they have a moderate to severe non-proliferative, they went for treatment like laser or injection to the district hospital. We have states, and then we have a state divided into districts, and then proliferative retinopathy was referred to a tertiary care center. So this was again published in the journals. And then they extended it, they found out 1 in 3 patients diabetes detected with diabetic retinopathy, 8.3% referred to secondary or tertiary ophthalmic care, and 38% of patients detected with diabetic retinopathy were in the age of 41 to 60, that is the productive age, and 99% of the patients were unaware in India that they had diabetic retinopathy. And it costed about $50 per patient for, no, sorry, 40 is... $5, I think, yeah. $5 per patient, and family, there were about 200 family health centers were screened, and this is one of their clinics in Kerala, which they did. So I'm now using this presentation to go and represent to the various governments, the public, and also non-governmental organization to do similar thing in India. In Mumbai, this is a map of Mumbai, which is a peninsula where we have 23 million people, and we plan to use this, and I'm trying to impress the government to buy 199 cameras and then use it. It will cost about $2 a patient for screening. So I follow my great leader, Swami Vivekananda, who said, they alone live who live for others, and rest are equivalent to a dead body. Thank you very much for the opportunity.
Pablo. Yes, Nati, and the device you use like a sleep lamp. So you only buy the camera, the device, so you can put the cell phone, or also all the table and the...
No, so this comes as a handheld, where the mobile phone is attached to an infrared device, which comes as a handheld. But I don't like the handheld because the examiner and the patient cannot be steady. So I have the slit lamp attachment, which is costing a few thousand rupees, lakhs, whatever. It's coming to 400,000 rupees. It's around $5,000. So another $250 will be the slit lamp attachment. So they can use it handheld, provided the examiner is steady and the patient also is steady.
And
they have an app in the phone where it has the AI app, which is offline. That means already there are about few 100,000 pictures of funders which is used as a source. So there is no need for internet, and the image is analyzed within the phone. So there is no need for sending it to center. And then you can also, when you don't have Wi-Fi, you can go to the place where the Wi-Fi and transfer all the images to the central server. So you can collect a lot of images, and the company also keeps it, and we also have the identity masked. So we buy the full system.
And it's made in India?
Yes, in Bangalore in India.
Okay.
And they also have official office in US, and I think in South America also, I don't know where, and also in Europe. They have CE certification and FDA approval. Anyway, I don't have any financial interest. They don't. No, but I love the technology because it's like we can say we had the richest man, Ambani, the Mukesh Ambani's father used to say that entire world in the pocket. That's what he created a Geo Mobile in India. Similar thing I said, the entire diabetic retinopathy in the pocket. That means in the mobile phone. You can detach it and keep it on the pocket.
Thank you. Thank you.
But I think we should do studies in... I just saw another camera with an Android camera from Brazil called Philcom. So I'm now trying to explore which is slightly cheaper than the Indian one, but I'm going to do a study. So my idea is to study various cameras available, and whichever country can adopt, and they should use it. And this is my dream for South America, India, and Africa.
Okay, great.
Yes.
To the audience, thank you so much for joining us. And we deeply appreciate all the speakers, this effort. And thank you, Dr. Thomas Ferner. This is the classroom of Klaus Forner. For support us, Universidad FASTA, Universidad Maimonides, and Consejo Argentino of Oftalmología, and all of us. Thank you so much.
And I think the YouTube will be there recording there all the time, right? So people can watch, and there are some questions in the YouTube I saw. Okay. Probably somebody should answer. And it was addressed to Takeshi, but I think we have to find a way how to address the question so that this particular thing can be a beginning for the prevention of blindness world over. Thank you.